Supported by a well-maintained campus area and qualified lecturers, both Indonesians and abroad make the university as a pleasant and conducive campus for teaching and learning process, as well as to develop students' interests and talents. Universitas Brawijaya opens the widest opportunities for prospective well-talented students, both domestically and abroad, including disabled. Currently, thousands of students earning scholarship from various institutions, both the government and private sectors. Universitas Brawijaya has 15 faculties and 2 programs of postgraduate and vocational. Supported by more than 140 professors and more than 2,000 well-experienced lecturers, researchers in teaching and research of various fields of science, technology, social, culture, and humanities, Universitas Brawijaya is ready to develop high-quality and international standardized educational process to produce graduates with high global competitiveness. In order to support students' competence and skills, Universitas Brawijaya provides diverse laboratories with sophisticated and modern equipment, including field laboratories, so the intense research culture continues to grow, indicated by innovative inventions that is useful for wide communities. Excellent scientific works and innovations are created by our academic communities as a means of education, research, and community service programs. As manifestation of its commitment to be a world-class entrepreneurial university, Universitas Brawijaya provides diverse infrastructures and equipment for diverse students' activities. This is an assurance of excellence and well-achieved learner profile produced. Lots of our alumni currently are in influential positions and have real contributions both in Indonesia and worldwide.
Dari pengajarnya dulu zaman saya kuliah banyak praktisinya, jadi saya ngerasa benar-benar kepake banget, bukan cuma belajar teori tapi prakteknya kita dapat.
Dari pengalaman kuliah yang paling berkesan ya karena saya anak organisasi, jadi ya di kepanitiaan, terus saya di sana belajar tentang kepemimpinan.
Dulu yang depan saya itu Fakultas Peternakan, sekarang udah berapa lantai udah ya, malah ada liftnya, dulu nggak ada.
Passionately, we, Universitas Brawijaya, will show the world an education with integrity and harmony at the forefront in science and technology development. Proudly and confidently, the graduates of Universitas Brawijaya are ready to welcome the future, building up the noble future.
Selamat datang di Fakultas Pertanian Universitas Brawijaya. Fakultas ini memiliki 4 jurusan, yaitu Jurusan Tanah. Bidang kajian di jurusan Tanah memfokuskan kelestarian lingkungan menuju terciptanya sistem pertanian yang berlanjut, manajemen kesuburan tanah dan ketersediaan sumber daya air bagi tanaman, serta manajemen sumber daya alam termasuk agroforestry dengan pendekatan ekologi ekonomi yang seimbang. Jurusan Budidaya Pertanian. Jurusan Budidaya Pertanian mempelajari dan menerapkan cara-cara peningkatan produksi tanaman serta perbaikan teknik budidaya tanaman secara berkelanjutan. Aspek yang dikaji yaitu genetik tanaman, fisiologi tanaman, penampilan fisik tanaman, dan kondisi iklim mikro dan makro di sekitar lingkungan tanaman. Jurusan Hama dan Penyakit Tumbuhan. Arah pengembangan keilmuan di jurusan HPT yaitu mengkombinasikan IPM, ICM, dan ISM untuk mewujudkan sistem pertanian berlanjut. Selain itu, jurusan Hama dan Penyakit Tumbuhan juga mendukung program nasional Go Organik untuk menghasilkan produk pertanian sehat yang bebas bahan kimia sintetik berbahaya. Jurusan Sosial Ekonomi. Fokus bidang ilmunya yaitu analisa perkembangan ekonomi pertanian nasional dan dunia literasi keuangan, pemasaran, dan analisa perilaku konsumen. Interaksi sosial dengan masyarakat pedesaan serta komunikasi efektif dengan pelaku agribisnis untuk mengembangkan pertanian yang berkelanjutan. Serta pasca sarjana. Kurikulum di pasca sarjana didesain khusus dengan kombinasi keilmuan berbasis kompetensi teori maupun terapan, sehingga sangat sesuai dengan kebutuhan para akademisi maupun para praktisi di sektor publik dan bisnis. Sejak didirikan pada tahun 1960, saat ini Fakultas Pertanian memiliki 11 program studi, dengan sebagian besar terakreditasi A oleh BAN PT, dan program studi di sarjana yang telah tersertifikasi AUN-QIA. Fakultas Pertanian Universitas Brawijaya memiliki 15 pusat kajian, 2 jurnal terindeks Scopus, 20 laboratorium keilmuan, 3 laboratorium lapang. Hutan Pendidikan UB Forest seluas 554 hektar, Agroteknopark Kebun Jati Kerto untuk teknologi budidaya komoditas pertanian dataran rendah, Agroteknopark Kebun Janggar untuk teknologi budidaya komoditas dataran tinggi, Kebun Percobaan Jatimulyo untuk kegiatan urban farming, serta memiliki ruang kelas untuk proses pembelajaran. Fakultas Pertanian bertujuan untuk menghasilkan lulusan pertanian yang profesional, kompeten, beretika, serta memiliki etos kerja yang tinggi sehingga mampu bersaing di tingkat internasional dan berperan aktif dalam pemberdayaan masyarakat melalui penerapan iptek bidang pertanian berlanjut serta manajemen agribisnis. Untuk mewujudkan hal tersebut, maka kegiatan akademik di Fakultas Pertanian difokuskan pada topik unggulan pertanian berlanjut di kawasan tropis.
Fakultas Pertanian Universitas Brawijaya berkomitmen untuk menjadi institusi pendidikan tinggi pertanian yang unggul, berstandar internasional, dan mampu berperan aktif dalam pengembangan iptek dan pengelolaan sumber daya alam yang berlanjut, serta pengembangan manajemen agribisnis. Untuk memperkuat jejaring pengembangan keilmuan, Fakultas Pertanian menjalin kerjasama dengan perguruan tinggi lain baik di dalam maupun di luar negeri dalam bentuk student exchange dan juga magang internasional. Selain itu, Fakultas Pertanian juga memberikan semangat kepada mahasiswa untuk berwirausaha serta kerjasama dengan lembaga internasional. Saat ini, Fakultas Pertanian berkonsentrasi untuk pengembangan pertanian terintegrasi dan lestari.
Honestly, I don't know where you are or where I am, but I'm not afraid of missing you. I'm strong enough to wait for you when the weather clears. So will our fears. Anything Anything holding us back will disappear. I'm coming for you, 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 I'm coming for you. Honestly, I don't know where you are or where I am, but I'm not afraid of missing you. I'm strong enough to wait for you when the weather clears. So will I feel anything holding us back will disappear. Coming
pandemic of COVID-19 has given an impact on many aspects of our life. The safety of food also become a concern of many researchers around the world. As food availability in regions and in the world can be threatened because of plant disease outbreaks. As we understand, the amount of food must be always available in accordance with the increase of the human population. Taking the anticipation and curative action against plant disease outbreaks is an urgent thing to be discussed and executed further. The understanding of agroecosystem, plant pathogens, beneficial microbes, and how plants become healthy become fundamental issues nowadays. In order to anticipate the outbreaks of plant disease and propose a novel method to control plant pathogen, we had a particular meeting among researchers and the field of phytopathology and stakeholders who are working in this field of area. This meeting not only involved the researchers from Indonesia, but also researchers from various countries. We hope through this meeting, we will have a sharing of newest knowledge, science, strategy, and technology on how to tackle plant disease to achieve sustainable agriculture in the area of Industrial Revolution 4.0, especially during the pandemic of COVID-19. pandemic of COVID-19 has given an impact on many aspects of our life. The safety of food
Recording in
also become a
progress.
concern of many researchers around the world. As food availability in regions and in the world can be threatened because of plant disease outbreaks. As we understand, the amount of food must be always available in accordance with the increase of the human population. Taking the anticipation and curative action against plant disease outbreaks is an urgent thing to be discussed and executed further. The understanding of agroecosystem, plant pathogens, beneficial microbes, and how plants become healthy become fundamental issues nowadays. In order to anticipate the outbreaks of plant disease and propose a novel method to control plant pathogen, we had a particular meeting among researchers and the field of phytopathology and stakeholders who are working in this field of area. This meeting not only involved the researchers from Indonesia, but also researchers from various countries.
Saya di Jogja.
Oh, kirain moderator ke Malang.
We hope through this meeting, we will have a sharing of newest knowledge, science, strategy, and technology on how to tackle plant disease to achieve sustainable agriculture in the area of Industrial Revolution 4.0, especially during the pandemic of COVID-19.
Excellency Dr. Insinyur Fajri Jufri MSI, Director General Indonesian Agency for Agriculture Research and Development, Ministry of Agriculture, Republic of Indonesia. The Honorable Professor Dr. Insinyur Muhammad Sasmito Jati MS, Vice Rector for Planning and Cooperation of Universitas Brawijaya Indonesia. The Honorable Dr. Insinyur Damanhuri SE, Dean Faculty of Agriculture, Universitas Brawijaya Indonesia. The Honorable Professor Dr. Insinyur Ahmadi Priyatmojo MSc, the General Secretary of Indonesian Phytopathological Society. The Honorable Professor Dr. Insinyur Abdul Latif Abadi MS, Chairman of Indonesian Phytopathological Society. The Honorable Professor Roger Koide, PhD from Brigham Young University, United States. The Honorable Professor Wei Changshan, PhD from National Taiwan University. The Honorable Dr. Insinyur Siwi Indarti, MP from Universitas Gadjah Mada, Indonesia. The Honorable Professor Nick Talbot, PhD from the Sainsbury Laboratory, Norwich, United Kingdom, and the respectable to all participants, International Conference and the 26th Congress of Indonesian Phytopathological Society. Assalamualaikum warahmatullahi wabarakatuh. Good morning to all honorable invited guests and participants. We would like to extend our warmest welcome to International Conference and the 26th Congress of Indonesian Phytopathological Society. It's an honor for me to be here in this occasion, so please allow me to introduce myself. My name is Khairunisa, and here I would like to guide the session for this event as the master of ceremony. The current industrial development leads to the 4.0 revolution, which involves changing patterns in the agriculture world. Agriculture that uses an integrated pest and disease management approach remains a very important approach to implement so that healthy crop yields can be obtained. The Indonesian Phytopathological Society is an association that accommodates experts and parties working in the field of plant disease science. Every 2 years, a congress is held as well as seminar to enliven the congress event. The seminar is international scale with important speakers for several different countries with a high level of publication citation in the field of phytopathology. This will be a great opportunity in a joint meeting to see scientific development not only in Indonesia but also in the world. International Conference and the 26th Congress of Indonesian Phytopathological Society is a part of the 61st anniversary of the Faculty of Agriculture, Universitas Brawijaya, Indonesia. It has a dream as the role of phytopathology in realizing healthy plants towards sustainable agriculture in the Industrial Revolution Era 4.0 that will be held in 29 until 30 of October 2021. On today, the first day, we will have an opening ceremony, keynote speaker presentation, and also the Indonesian Phytopathological Society Congress. And for tomorrow, the second day, there will be a paper presentation and also the second ceremony. For the first session, there will be a report from Secretary of Organizing Committee of International Conference and the 26th Congress of Indonesian Phytopathological Society to Mr. Muhammad Akhid Shibli, SPMP, PhD. Time is yours. Good morning, Mr. Shibli. Can you hear my voice well there? I think you still mute your Zoom. Okay. How about now? Can you hear my voice well there?
Yeah. Thank you, Nisa.
Okay.
Thank you.
You can start whenever you are ready. Time is yours.
Thank you so much, Nisa, for the opportunity in this important meeting. So everyone, good morning. I'm Muhammad Shibli, and I would like to share the report of the organizing committee. Can everyone hear my voice? All right. So allow me to prepare this PowerPoint first. I'm sorry, a little bit of trouble here. All right, so let me prepare first. Okay. Good morning, everyone. Thank you so much for these important meetings that we can hold together in this morning. So first of all, I would like to thank you very much for the attendance, the Honorable Professor Sasmita, Vice Rector of Universitas Brawijaya, the Honorable Dr. Fajri Jufri from the Ministry of Agriculture, and also Professor Abdul Latif Abadi, the Chairman of the Indonesian Phytopathological Society, and also welcome to Professor Ahmad Wiatnojo from Universitas Gajah Mada, also as the General Secretary of the Indonesian Phytopathological Society. So in this morning, I would like to give a little bit of story about this event. This event able to be held today in this morning. because we have a collaboration between the Indonesian Phytopathological Society, the committee itself, and the Department of Plant Pathology and Disease, Faculty of Agriculture, Universitas Brawijaya. And we have made this poster from this early year. We have been published from March, and we have theme the role of phytopathology in achieving healthy plants towards sustainable agriculture in the era of the Industrial Revolution 4.0. And in this important meeting, we have 2 days from 29th until 30 October 2021. And we have successfully communicated and discussed further with the invited speakers and also the keynote speakers. From the Ministry of Agriculture, Dr. Fajri Jufri, and also Professor Rokrede. He will talk about the ecology about endophytic fungi. He's professor from Perhimpunan University. And also Professor Nick Talbot, Group Leader and TSL Executive Director from Nick Talbot Group. He will elaborate more about the Magna Proteoryzae, rice blast fungus. And this is, as we know, that this become the most important, I can say, pathogen for the party plant. And also we have Prof. Wei Chiang-shen from National Taiwan University. He will also talk about rice blast fungus in Taiwan. So hopefully we will have new insight from Group Taiwan. In Taiwan, I mean. Okay, the next is Dr. C.V. Indarti. He will talk about the management of parasitic nematode toward sustainable agriculture. So we have published this event in the website of our society, also the Instagram. We also made the video promotion for this event so everybody can see, even the speakers from the crowd can see this video. Some of group I have shared for this video file so they can share among the group. We also have accepted about 106 abstracts. From this graphic, we can see that group from Mycology and Plant Disease Management in Indonesia is still the highest number until now. So from other group, we need more effort, more spirit to conduct more research. Okay, for the next, I can say that this event held in one of beautiful university, Universitas Brawijaya. And because this pandemic, we are not able to see each other directly and not able to talk directly. Actually, I really want to talk with you, all of you, directly, shake your hand, talk without mask here in Universitas Brawijaya, so we can enjoy the view here. And also, because I'm so confident that Malang City has a very beautiful view, not only in the center of city, but in the around of Malang City, Malang Prefecture, we have fantastic view here. You can see the Brahma Mountain here. But also don't forget, we also have the best cuisines, I can say. So please visit us. Can you see? We have the best cuisines ever. This is hot training, I can say. And also nasi pecel kawi. So you can enjoy this nasi pecel kawi near Universitas Brawijaya. While you're enjoying the view of Malang City, you can eat this together with me if you want. And the last one, don't forget about bakso and satay. So, special thank you also for the sponsor and support, Universitas Brawijaya, and also PTBC International, Petrokimia Kayaku. In this moment, I would like to say, let's enjoy the international conference and the 26th Congress of IPS. Thank you very much. Sugeng rawuh sedaya Bapak Ibu. at Universitas Brawijaya. Thank you, Anissa.
Well, thank you, Mr. Muhammad Akhid Shibil SPMP PhD for the report. And ladies and gentlemen, of this we will have a special traditional dance performance for opening ceremonial from Universitas Brawijaya. Sekar Taji Dance is a dance that tells about the struggle of Raden Panji Asmorobangun to maintain his love for Dewi Sekartaji. Dewi Sekartaji character is kind, gentle, and holy nature, while Raden Panji Asmorobangun as the central character is handsome, dashing, loyal, and refined, and unpretentious nature. And this is Sekar Taji Dance. Once again, please give virtual applause for the performance. Well, distinguished guests, for the next session, we will listen to a remark from Director General Indonesian Agency for Agricultural Research and Development, Ministry of Agriculture, Republic of Indonesia, to Dr. Insinyur Fajri Jufri, MSI. Time is yours.
Honorable Rector of Brawijaya University, Professor Novil Hanani and his staff, Chairman of Indonesian Phytopathological Society, Professor Dr. Insinyur Abdul Latif Abadi, and all the members, invite speaker, honorable guests, and all participants. Assalamualaikum warahmatullahi wabarakatuh. Good morning and greeting to all of us. First, I would like to thank the presence of Allah Almighty for the abundance of grace and the joy that all of us can still gather in this conference today without any barrier and all in good health. I would like to say congratulations to Indonesian Phytopathological Society for organizing this wonderful international conference with the theme, The Role of Phytopathology in Achieving a Health Plan Towards Sustainable Agriculture in the Era of Industrial Revolution. This theme is very important for the development of agriculture sector in Indonesia. Distinguished guests, ladies and gentlemen, during the COVID-19 pandemic, agriculture has proved this sector is the backbone of Indonesian economy. For example, in the first 3 months of 2021, agriculture contribute 60.24% to GDP. Also, the agricultural sector is the leading contributor to national export, even the weakening of the world economy due to COVID-19 pandemic. In 2020, the export value of agricultural commodity reached 451.55 trillion rupiah, or grew by 60.5% compared with 2019. But the plantation subsector contribute 91% of the export. Recently, on 14 August 2021, Mr. Presiden Jokowi released agriculture export product simultaneously from 74 seaport and airport in 17 provinces with 61 destination countries. The export product released on the occasion amount 7.29 trillion rupiah. Ladies and gentlemen, the agriculture development in Indonesia face many issues covering social, economic, and environmental aspects that lead to the sustainable issues. Even though agriculture expansion is considered one of the main causes of environmental externalities such as deforestation, land use change, and greenhouse gas emission, which contributes to climate change, agricultural production is also highly affected by the change in climate pattern. Many studies show that changing climate highly contributes to increase of pests and disease attack, which cause a significant decrease in agricultural production. The International Plant Disease Epidemiology workshop in 2005 predicted that pests, including plant pathogens, cause losses in pre-harvest yield in the range of 20 to 40%. An estimate of post-harvest losses are higher than 10 or 20%. In addition, the use of chemical pesticide in order to control pests and disease attack has been a big concern regarding health and sustainable issues. Therefore, the contribution of phytopathologists and Indonesian Phytopathological Society to overcome the issues become very important. Ladies and gentlemen, in the era of Industrial Revolution 4.0, artificial intelligence and big data have been seen as a new solution for many problems, including in agriculture. This technology allows us to implement precision agriculture, such as for monitoring microclimate, diagnosing plant health status, monitoring plant growth, and others, effectively and efficiently. Hence, I see that technology 4.0 is also the opportunity to be developed to address the plant disease problem in Indonesia, which is also important to increase the productivity, quality, and sustainability of agricultural production. Ministry of Agriculture and ARD have a commitment to support the creation of technology innovation in agriculture, including 4.0 technologies. Our Minister, Dr. Syahrul Yassin Limpo, on many occasions always strengthen the importance of the use of 4.0 technologies such as artificial intelligence, Internet of Things, big data, and others in agriculture. To follow up on the instruction, we need to create the technology here in Indonesia and have the support and contribution for the Indonesian Phytopathological Society, especially in the field on plant diseases. In conclusion, I would like to strengthen the important role of phytopathology on agricultural development in Indonesia. I really appreciate and believe that this conference and congress will result in some important idea in achieving healthy plan toward sustainable agriculture. I wish you every success during the conference. Enjoy your conference. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Thank you to Dr. Insinyur Fajri Jufri MSI for the remark. And for the next session, we will listen to a remark from Chairman of Indonesian Phytopathological Society, to Professor Dr. Insinyur Abdul Latif Abadi MS. Time is yours.
Bismillahirrahmanirrahim. The Excellency Director General of Indonesian Agency for Agriculture, Research and Development, Ministry of Agriculture, Republik Indonesia, Dr. Fajri Jufri. The Honorable Rector of Universitas Brawijaya, in this occasion, will be represented by Professor Dr. Sasmito Jati, the Honorable Dean of Faculty of Agriculture, Universitas Brawijaya, Dr. Daman Huri, the Honorable General Secretary of IPS, Professor Ahmadi Priyatmojo, PhD, the Honorable keynote speakers, all distinguished guests, participants of international conference and the 26th Congress of the Indonesian Phytopathological Society. Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. It is such a great pleasure to welcome you all at this important meeting. I also want to welcome all of you at Malang, even though at this point we can only meet virtually. But this will not reduce our spirit and enthusiasm of all of us in carrying out this activity. On behalf of the organizing committee of the conference, allow me to deliver several points. The Indonesian Phytopathological Society is an association that accommodates experts and parties working in the field of plant disease science. Every 2 years, a congress is held as well as a seminar to enliven the congress event. The seminar will be held in 2021, will be broadcasted internationally with keynote speakers from several countries with a high publication citation rate. This year, our committee tried to raise the theme, the role of phytopathology in achieving healthy plants towards sustainable agriculture in the era of industrial revolution 4.0. The theme was raised as an effort made to add insight and practices that need to be done in creating healthy plants. The Indonesian Phytopathological Society accommodates to share knowledge related to the role of phytopathology itself in creating healthy plants in the context of sustainable agriculture in the era of industrial revolutions 4.0. This year, there were 2 sessions of activities, namely international seminars and the Congress itself. To add insight to all participants, especially the Indonesian people, we invited several international speakers from United Kingdom, United States of America, Taiwan, and also Indonesia, who I believe will bring new insight to development of plant disease science. Ladies and gentlemen, representing the organizing committee of the conference, I would like to express my deepest gratitude to everybody taking a part in this conference. Keynote speakers, participants, parallel presenters, steering committee, organizing committee, and also Brawijaya University, sponsors, and also everyone else. Even though we, the committee, have tried our best effort to make a better conference this year, I believe that there remains some inconvenience. For that, we do apologize. That would be the end of my speech. One more time, deep from my heart, thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Thank you, Professor Dr. Insinyur Abdul Latif Abadi MS for the remark. And for the next session, we will listen to a statement from Vice Rector for Planning and Cooperation of Universitas Brawijaya Indonesia, which will officially open the international conference and also the Congress of Indonesian Phytopathological Society. To Professor Dr. Insinyur Muhammad Sasmito Jati MS, time is yours.
Assalamualaikum warahmatullahi wabarakatuh. Good morning to all of you. I hope all of you will find and will happy. It is my pleasure for me to standing here to give some speech, short speech, to opening the conference with the title, The Role of Phytopathology in Achieving Healthy Plants Towards Sustainable Agriculture in the Era of Industrial Revolution 4.0. Firstly, it is my Honorable Professor Ahmadi Priyatmojo, who stay here in this campus. Also, my Honorable Professor Dr. Insinyur Fajri Jufri, Professor, the keynote speaker, Professor Roger Koide from Brigham Young University, Professor Wei Chiang Shen from National Taiwan University, Professor Siwi Indarti from Gajah Mada University, Professor Nick Talbot, PhD from the Sainsbury Laboratory, Norwich, UK. It is our pleasure to stay here, to standing here on behalf of Brawijaya University. I hope this conference, international conference, will be organizing well. And as I know, even I'm not an agriculture scientist, but I know about the ecology. Indonesia is very interesting place for discussion about phytopathology, because we living in rainforest, tropical rainforest. I think in the worldwide tropical rainforest, it is many pathophysiology that have spread easily, not just the effect in plant, but also in human. As we know, now we are under pandemic. It is very easy to spread out many viruses in Indonesia. But we believe in this occasion, you will discuss about many things interdisciplinary, and also all of you will solving the problem in terms of pathophysiology, in terms of plant pathology. And I hope in this occasion also we can make some solution to improve our agriculture production, and hopefully it will be giving a big impact to Indonesian in terms of agriculture production. Anyway, now in terms of pandemic era, I think if we think about the discussion, if we think about the conference in terms of online, I think now we have many experience to facing our era in terms of Revolution 4.0. But even this era we can solve the problem with online communication, online conference, online something like that. I hope in next year or next meeting we will be real meeting. That's mean we hope to pray to the gods that the pandemic will be finished. I hope, amen. And then I hope in this conference we will make our students much more have face-to-face meeting, and in that occasion, if I will be more have many experience, many communication and something like that. Plant pathologists face a challenge of responding to this problem, being contributing to more inclusive forum for multidisciplinary research, recognizing that the impact will be depend not only on advancement in the plant pathology that I have taught before, but also in interaction more broadly with other agriculture and ecological sciences, and with national and global needs. In this conference, as one of many endeavor to realize UB vision and mission to become a world-class research university, this year the conference also celebrating the 61st Faculty of Agriculture University. It is a part of our concern about the role of science and technology in improving Indonesia national productivity in competitiveness. Hopefully, we'll be able to contribute substantially to the country advancement in the future. Last but not least, I just want to say, to open this conference with the saying, Bismillahirrahmanirrahim, in the name of God, we officially open the meeting. Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Professor Dr. Insinyur Muhammad Sasmito JTMS for the statement. And now International Conference and the 26th Congress of Indonesian Phytopathological Society is officially open. Ladies and gentlemen, by the grace of our God, we all are allowed to gather here in such a beautiful moment and place. And for such a blessing, let us pray together. And the prayer will be led by Mr. Ahmad Muafiq Saleh, SOCBSI. And time is yours.
Assalamualaikum warahmatullahi wabarakatuh. Ladies and gentlemen, let's all pray to Allah, Lord of the world. Allow us to read the prayer in Islam. Bismillahirrahmanirrahim. Alhamdulillahi rabbil alamin. Wassalatu wassalamu ala ashrafil anbiya wal mursalin. Sayyidina wa habibina Muhammadin sallallahu alaihi wasallam. Allahumma salli ala sayyidina Muhammad. Sahil wa yassir ma ta'assar. Allahumma salli ala sayyidina Muhammadin. Allahumma ja'al jam'ana hadha jam'an marhuma wa tafarruqana min ba'dihi tafarruqan ma'suma. ولا تجعلنا منا شقيا ولا محروما. Ya Allah, we ask you to make this meeting useful for life, contribute to science, and become the path of our happiness in the hereafter that will take us to heaven. Ya Rab, make our greeting is blessed catering and our spiration after this us a blessed and protected parting. And do not make this open us and those who are with us virus and remove from obtaining mercy. Rabbana taqabbal minna innaka antas samiul alim wa tub alayna innaka antat tawwabur rahim. Allahumma rabbana zalamna anfusana wa illam taghfir lana wa tarhamna lanakunanna minal khasirin. Sahil jami'a umurana, sahil jami'a umurana, sahil jami'a umurana. Allahumma altuf bina fi taysir ful kulla asir fa inna taysir kulla asir fid dini wad dunya wal akhirah. Rabbana atina fid dunya hasanah wa fil akhirati hasanah wa qina adzaban nar. Wa sallallahu ala sayyidina Muhammadin walhamdulillahi rabbil alamin. Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Wassalamualaikum warahmatullahi wabarakatuh. Thank you, Mr. Ahmad Muafiq Saleh, SOSBSI, for leading the prayer sessions. Well, ladies and gentlemen, we finally get to the main session of this event, International Conference and the 26th Congress of Indonesian Phytopathological Society. As we need to inform you that the participant of the seminar will be divided into two sessions. The first session will be held before the break session, who will be delivered by Professor Roger Koide, PhD, from Brigham Young University, United States, and then Professor Wei Chiangshan, PhD, from National Taiwan University. And for the second session, will be held after break session, will be delivered by Dr. Insinyur Siwi Indarti, MP, from Universitas Gadjah Mada Indonesia, and also Professor Nick Talbot, PhD, from the Sainsbury Laboratory in Norwich, United Kingdom. The first session will be led by our moderator, and I will be directly reading the curriculum vitae. In 1985, he has completed his undergraduate studies at Universitas Jenderal Sudirman, Indonesia, majoring in plant pests and disease. In 1989, he has completed his master's studies at Universitas Gadjah Mada, Indonesia, majoring in phytopathology. In 2000, he has completed his doctoral studies at Wageningen University, Netherlands, majoring in plant science. And his research focus is about biological control. He has a lot of track record in making scientific publication and research both on national and international scale. And he also invited to be a speaker at the national and international seminar. And now let us welcome Professor Insinyur Lucas Susanto, MS, PhD, from Faculty of Agriculture, Universitas Jenderal Sudirman, Indonesia.
Okay, thank you, the moderator. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. Good morning, ladies and gentlemen, and good afternoon, Professor Koide. Great opportunity to lead the discussion, and today we have a special guest and honorable guest, and hopefully you are in a good health. And the first guest and the honorable guest who will give us a new knowledge and perspective in plant pathology, in plant disease. And before I deliver this screen to the first guest, I would like to remind you all to mute the the speaker. And when you have a question, you can write down in the chat public. Now, I would like to ask the committee to share the first guest curriculum vitae, Professor Koide. Please, the committee. Can you hear me? Okay, Professor Roger T. Koide. His PhD from Botany from University of California, Berkeley, and BA in Zoology from California. And he has a professional experience in many expert and professional experience in Japanese Society for the Promotion of Science Fellowship, Fulbright Senior Scholar Award Australia, and another. And now to Professor Koide, time and screen is yours.
Thank you very much. I'll just share my screen then.
Yeah.
And can everybody see this all right?
Yes.
Okay. Well, thank you very much for the opportunity to be here today. I'm grateful that I could share some hopefully interesting things with you. And I very much appreciate the prayer beforehand. I could always use help from God and as I give this seminar. And thank you to the committee and everybody who's been involved in inviting me. Thank you very much. So I'm going to be talking about the ecology of endophytic fungi. Can you hear me okay? No problem?
Yes, Prophet Koide.
Okay, very good. Before I begin with the talk on the endophytic fungi, I just wanted to go over some general principles of symbiosis because anybody who studies plant pathogens is a student of symbiosis. And I hope everyone understands that symbiosis is not the exception in nature, it is the rule in nature. All eukaryote species form intimate associations with other species and especially microbes. Therefore, eukaryotes are not autonomous individuals. They don't act by themselves. They are what we call holobionts, which consist of the eukaryote and its community of symbiotic microorganisms. For example, coral polyps form symbioses, very intimate symbioses with photosynthetic algae. Plants form symbioses with mycorrhizal fungi. And vertebrates form intimate symbioses with gut bacteria and fungi and protists. The interesting thing is the host organism acquires new traits from its symbiotic microbes. And as a consequence of these new traits that are determined by the symbionts, the host occupies altered niche space. Its niche has been changed. For example, coral polyps acquire photosynthesis. Plants acquire enhanced phosphate acquisition. And vertebrates, like humans, acquire the ability to convert food, to produce vitamins, and to train our immune systems that otherwise would not be possible without the microbes. Also, symbiosis offers an alternative to neo-Darwinian adaptation. There are ways of adapting to new circumstances through symbiosis and not just through neo-Darwinian evolution. Let's consider first these coral polyps. Let's say for now they do not have symbiotic algae growing in them. They're not photosynthetic. And if these polyps can't obtain sufficient nutrition from the water, it would be obviously advantageous for them to evolve photosynthesis. But because they do not have the genes necessary for photosynthesis, it's essentially impossible for them to evolve intrinsic photosynthesis, for them to become photosynthetic themselves. But photosynthesis can be acquired through symbiosis. In other systems, it may be possible for both intrinsic or neo-Darwinian and symbiotic adaptations to evolve simultaneously. But in cases where genetic, structural, or developmental constraints make the intrinsic adaptation impossible, symbiosis may save the day. It's a different way of producing an adaptation. And one advantage of the symbiotic approach to adaptation is the possibility of swapping symbionts, changing the symbionts that you're associating with. And that makes further adaptation very simple and very rapid. For example, this is a very short story about the desert wood rat, and it maintains populations in the 4 major Western deserts. So it lives in the Great Basin, and this is where I live. I live in the Great Basin. It lives in the Mojave Desert, and the Sonoran Desert, and the Chihuahuan Desert. And in the 3 southern deserts, we have this plant called creosote bush, and it forms part of the diet of this wood rat in the Mojave and the Sonoran and the Chihuahuan deserts, but it doesn't grow in the Great Basin. The wood rats in the Great Basin, if you try to feed them creosote bush, they die because the bush has the leaves are coated with a toxic resin, poison. But the wood rats living down here and here and here, they can tolerate this as a food source. Initially, people thought that the tolerance to the resin in the plant was an intrinsic trait of the wood rat. It was determined by the genes of the wood rat. But more recently, it was shown that detoxification of the creosote bush resin is due to microbes living in the gut. And what that means is if you take the gut microbes out of one mouse that can tolerate the resin and put them in the gut of a mouse that cannot tolerate the resin, now suddenly it can tolerate the resin. So symbiont swapping can lead to rapid adaptation, even in the absence of neo-Darwinian evolution. So you see, symbiosis is not only pervasive, it occurs everywhere, but it is very, very important for the adaptation of organisms, plants and animals of all kinds. So the fundamental unit in ecology is not really the individual, like the individual coral polyp, but the holobiont, the coral polyp with all of its symbiotic microorganisms. The existence of holobionts also makes the species concept very difficult to apply. For example, many sap-sucking insects, those that feed on the phloem sap of plants, they have bacteria associated with them very intimately. The bacteria grow inside insect cells, and those insect cells are organized into organ-like structures called bacteriomes. So here's a bacteriome here. And here is a fluorescence microscopy photograph of portions of the bacterium. These little green things inside this host cell are bacteria of one kind, and the red ones are bacteria of a different kind. The bacteria are transmitted vertically from parent to offspring. The bacteria cannot live independent of the insect. The insects cannot live independent of the bacteria because they depend on the bacteria to produce certain amino acids and vitamins. So this symbiosis is really intimate, and neither of the organisms can live independently of the other. So this suggests that the species concept is a very difficult one in some symbioses like this. So the fundamental unit in evolution is not the species, but the population of holobionts. Plants form a number of symbioses with various microbial species, and these microbial species can have profound effects on the niche occupied by the plant, and thus on the ecology and the evolution of the holobiont. There's just a picture of some types of plant symbioses with microorganisms, arbuscular mycorrhiza, the legume rhizobium symbiosis, and this pathogen, Armillaria melia. Well, today I'm going to be focusing on symbiosis between plants and endophytic fungi. This is just a photograph of several isolates of endophytic fungi that we isolated from Gamble oak leaves. Some endophytic fungi are famous. For example, these endophytic fungi from Taxus leaves and bark produce Taxol, which is an important anti-cancer chemotherapy agent. As it turns out, all terrestrial plants are colonized by endophytic fungi. Their leaves are colonized, their stems, their flowers, their roots, just about all the parts of a plant are colonized by endophytic fungi. Well, what are endophytic fungi? Endophytic fungi, I want to define them as simply fungi that live within plant tissues, not within plant cells, but within plant tissues. And we don't want to define them by their function, only by their habitat. And we don't define them by their function because their functions are quite variable. Some endophytic fungi confer advantageous traits on their host. Therefore, these are thought to be mutualistic. For example, this fungus growing in cool season grasses, Epicloe, is capable of producing alkaloid compounds that deter herbivores. So when plants are colonized by Epicloe, then the plants become less susceptible to herbivory. This one, Paraphoma and Guaiamonomyces, these two species can increase heavy metal tolerance in certain plants. And this one, another Epicloe, can increase stress resistance in plants, drought resistance and heat stress resistance. And the top photographs, this is without the endophytic fungus, and the bottom four photographs with the endophytic fungus. And you can see the controls here, and then heat stress without and with, drought without, drought with the endophyte, heat and drought without the endophyte, and heat and drought with the endophyte. So these can increase stress tolerance. But while some endophytic fungi are mutualistic, some may be pathogenic, depending on the circumstance. For example, we have what is called age-dependent expression of pathogenesis by endophytic fungi. When leaves are young, the pathogen, the fungus may not be pathogenic, but as the leaves begin to senesce, then the expression of pathogenesis may occur. Also, some endophytic fungi are pathogens in some host species, but not in others. So it may be a mutualistic fungus in one plant species, it may be a pathogenic fungus in another plant species. And as we have shown, and as others have shown, many endophytic fungi are saprotrophic, that is, they are decomposer fungi. So we say they have priority over other saprotrophs for resources in litter because when the plant tissue dies and becomes litter, it is already colonized by the endophytic fungi. And if the endophytic fungi then become saprotrophic, they have first access to the resources in that litter. So the term endophytic only describes the habitat of the function, not a habitat of the fungus, not the function, because the functions are quite variable. Variation in function may reflect variation in the evolutionary paths resulting in endophytism. There may have been multiple evolutionary pathways that lead to endophytism. And a way of describing these multiple pathways might go as follows. Let's think about a party and you've invited several guests to your party. When a guest arrives early, she will have access to the food before anyone else arrives. And decomposer fungi, they normally colonize plant tissues only after they die. But some decomposer fungi may have evolved to colonize plant tissues prematurely. They may arrive before the rest of the party arrives. That is, while the tissues are still alive. Like the guest who arrives early, when the plant tissue dies, these fungi have access to resources in plant tissues before the decomposer fungi arrive, the other decomposer fungi. So one way to evolve into endophytes is for decomposers to begin to colonize tissues before they're a litter, arriving early. Another pathway to endophytism might be this. One guest may linger after all the other guests have gone home in the hope that she can have a second or third round of food. Pathogenic fungi colonize living leaves, normally causing disease or death, but some pathogens may have evolved to linger a little longer, to limit pathogenesis in order to keep access to a living source of nutrition for quite some time. Sorry about that. And the third way, another way of arriving at endophytism may be as a pre-adapted mutualist. The best friend of the person holding the party may come early to the party to help prepare for the party and may stay afterwards to help wash the dishes and clean the house. And that is a mutualistic friend, someone who is beneficial. And some fungi may have been pre-adapted to becoming mutualistic. For example, if fungi produce alkaloids and those alkaloids deter herbivores, then it may be selected for by the plant to colonize it, to confer some advantage like herbivory resistance. Now, probably there have been multiple evolutionary pathways to endophytism, so it's not surprising, therefore, that endophytic fungi have a multiplicity of functions. And as I said earlier, we've been interested in saprotrophy. How many of these actually become decomposer fungi or are decomposer fungi? And we first got an inkling about this in an experiment we performed a few years ago on Quercus gambellii, this oak that lives in Utah. And we isolated fungal DNA, or we purified fungal DNA from living leaves, shown over here, as well as leaf litter that had recently fallen to the forest floor. So shortly after it fell, and then over the course of 6 months. And this shows the relative frequency of various fungal taxa over time. The first date is in living leaves, and then the subsequent 3 dates are in litter as it decomposes. The first 3 panels, A, B, and C, show that these taxa were essentially non-existent in living leaves, and they became more frequent as the litter decayed. So these are probably non-endophyte saprotrophs, non-endophyte decomposers. This fourth panel here, over here though, all of these fungi were present, had a significant presence in living leaves, and maintained a presence in the decaying leaves. So we believe these are endophytes that are also saprotrophic, that are decomposers, but have colonized living leaves. So in order to determine whether endophytic fungi truly are saprotrophic, we did a subsequent laboratory study using cultures of the various isolates we obtained from Quercus gambellii leaves. And in these darkly stained cultures, the only source of energy in those cultures is ground up litter, leaf litter. So if they grow substantially on this material, we think they must be saprotrophic. They can obtain energy from leaf litter. This is a plot of saprotrophic capacity, or a measure of how well they can grow on leaf litter. And you can see of the 40 or so isolates here, all of them except these first 2 had significant saprotrophic capacity. There is a lot of variability among isolates and their saprotrophic capacity. Nonetheless, they have all been able to, with the exception of these two, all been able to obtain energy from leaf litter. And again, the ecological significance of this is these are decomposer fungi, but they're already in the litter when the litter falls to the forest floor because these are endophytic fungi. They're there first, so they have priority over other decomposer fungi. Another thing that we've been interested in is the structure of fungal communities, endophytic fungal communities. Some plants have very simple endophytic fungal communities. For example, the endophytic fungal communities in cool season grasses are very often very, very simple. Sometimes they comprise only a single species of fungus. But other species of plants may contain multiple species of endophytic fungi. In this study of ours of 6 different plant species listed up here, these are the plant species. This is a list of the groups of endophytic fungi by order. And you can see that some of these are all very, very complicated fungal communities. Now, function varies among species of endophytic fungi. So the function of the community is going to be determined by the structure of the community. So we want to know what structures these fungal communities, what determines the composition of an endophytic fungal community. And so I have this simple model here. Dispersal, climate filtering, host filtering, and competitive interactions. So dispersal is what determines what species are available at your site, what species were actually able to disperse to your site. That's the immigrant pool. Now, not all of the immigrants may be capable of living in that climate. So we have this climate filter here. And so the species that actually can survive in this particular site are those that are adapted to that climate, a smaller subset of this immigrant pool. And then not all of these that are climate adapted may be able to colonize the host of interest. And so there's a host filter here. And this group of species here is what makes it past that filter. And then, of course, competitive interactions, if they exist among these species, may result in the realized community. So we're going to look at each of these in turn to see which are important, which are not important. First, the role of dispersal. This is an aerial view of where I live. This is Provo, Utah here. This is Springville, Utah. This is the town where I live. And this is the Front Range. And you can see it's running north and south. And there are all these canyons that cut across the range in an east-west direction, which means each of these canyons has a north-facing slope, or a south-facing slope, I should say, and a north-facing slope. So we collected leaves from Quercus gambellii growing on south-facing slopes and north-facing slopes from each of 4 different canyons here in Utah. This is the result of a Mantell test. And a Mantell test is really just a correlation method to determine the correlation between geographic distance and community structure. We did this Mantell test for south-facing sites and for north-facing sites. And you can see here, both of them show that the Mantell test is significant, which means the distance was significantly correlated with fungal community structure. And that's consistent with the fact that dispersal limits fungal community assembly. This is true in both north and south. But if you look here, you can see here, this is the R squared. And so 8%, 3%, these are not large effects, even though dispersal seems to be significant. It doesn't seem to have a tremendous impact, 8%, 3%, on the structure of the fungal communities. What about the role of climate? The same study could be used to examine climate because the climate on a south-facing slope and the climate on a north-facing slope are quite different from each other. Here's a close-up of one of those 4 canyons. Here's the south-facing slope. It's warmer and drier. Here's the north-facing slope. It's cooler and wetter. And when we collect leaves from Quercus gambellii here and leaves over here, we can test for the effects of climate. These are ordination plots, and in an ordination plot, when two dots are close together, that means the communities, the fungal communities, are similar, and when they're far apart, the fungal communities are more different. You can see that for each of these 4 canyons, the north-facing, the communities and leaves from the north-facing plants group together, and the communities from the leaves on the south-facing plants group together, and they're significantly different from each other in all 4 canyons. And this suggests that climate plays a significant role in the structuring of these endophytic fungal communities. Let's see, am I supposed to be monitoring the chat? There's some questions. Yeah, I'll have to get to those questions later, I guess. Sorry about that. Let's see. So let's look at host filtering. We did another study about 50 miles west of here in the desert. This is the Great Basin Desert. And we studied 6 different plant species. Bromus tectorum, Elamis elamoides, these are grasses. Artemisia tridentata and Chrysothemis depressus, these are members of the Asteraceae, or the sunflower family, and Atriplex canescens, and this is being covered up, this mustard, Alyssum elysoides. These 2 are not mycorrhizal, and that's going to be significant. These 4 species are mycorrhizal. They can make mycorrhizal roots, but these cannot. These 2 are in the grass family, the Poaceae. These 2 are in the Asteraceae, or the sunflower family, as I said. And these are both non-mycorrhizal, but in different families. This is the Amaranthaceae, and this is a Brassicaceae. This is a phylogenetic tree of the orders, plant orders, and you can see the sunflower, members of the sunflower family are up here. This is the first non-mycorrhizal species. It's in the Brassicaceae. Atriplex is in the Amaranthaceae. It's also non-mycorrhizal. Bromus tectorum and Elamis, these are both grasses, and they're down here. Okay, now this is another ordination. And again, when things are close together, the communities are similar. When they're far apart, they're more different. Now, the 2 grass species, Elamis and Bromus, their symbols are enclosed within the green and the blue ellipses. And you can see they're very, very similar. The 2 grasses have very similar communities of endophytic fungi. The Chrysothemis and Artemisia are the sunflower family, and those 2 have very, very similar communities, endophytic fungal communities, but those communities are different from the grass communities. And finally, these are non-mycorrhizal in the Amaranthaceae and in the Brassicaceae. So variation in endophytic fungal communities appears to be attributable to plant family and to mycorrhizal status. But we didn't know how much of that was simply due to dispersal limitation, because the vegetation in this community is a little bit clumped. So in order to look at community structure independent, due to host filtering, independent of dispersal limitation, we had to perform a separate study in the greenhouse. And this is that study here. So these plants, these are all Bromus tectorum plants, and they're growing under very controlled conditions with various sources of inoculum. Now, the inoculum sources were either Elamis elamoides litter, or Bromus tectorum litter, or Chrysothemis litter, or Artemisia litter, or soil. This is all the inoculum sources. So you can see this is, again, another ordination plot. These are the communities, fungal communities, from the various sources of inoculum, and you can see they're quite different from each other. But when you look at the fungal communities in the tissues of Bromus, of the test plant, that's these darker symbols here. They are very similar to each other, irrespective of the inoculum source. That's true for the leaves, that's true for the roots. So leaf and root fungal communities of Bromus tectorum deriving from disparate inocula were similar despite large variation in inoculum fungal community structure, which means the host filtering effect is strong. So what determines the composition of an endophytic fungal community? Let's see. I can't see my whole screen. Here we go. I've got to get rid of this. Okay, so dispersal is weak, abiotic filtering climate is moderate strength, and host filtering is very strong. We didn't test competitive interactions. It's probably weak because these fungi are what we call class 3 endophytic fungi, which means they don't grow much inside their host plants, and so it's not likely that they interact with each other very, very greatly. Okay, agricultural applications. I said earlier that there are a number of mutualistic benefits associated with some of these associations. So for example, some of these fungi produce alkaloids which can deter herbivores. Some of these fungi increase heavy metal tolerance or tolerance to heat and tolerance to drought. And so of course, there's a lot of interest in using these fungi in agriculture, but we have to be careful. While there are some microbes that have been commercialized and may be useful as inoculants, for example, Rhizobium or Trichoderma, these don't always work. And one reason why is because, one reason these are not always effective is because there is going to be competition between the inoculant taxa and the indigenous taxa or the resident taxa. And unless the inoculant taxa are very, very good at invading an intact community, they're not going to be able to colonize the plant species of choice. In fact, it's so difficult to inoculate some plants that you have to grow them under sterile conditions like this in order to inoculate them with some endophytes. The other problem is, if they are able to overcome competition from indigenous taxa, they have to be very, very good invasives, which means they're very, very strong, very, very competitive, potentially invasive. That means they can, let's say you're trying to inoculate corn, but you have soybeans growing next to the corn, you may inadvertently inoculate the soybeans. It may be a mutualist in the corn, but maybe a pathogen in the soybeans. Or if you're trying to inoculate soybeans and it's a mutualist in soybeans, if your next crop the next year is wheat and it's a pathogen in wheat, well, then you're out of luck then. So it's environmentally risky also to use some of these inoculants. So to summarize quickly here, all eukaryotes are symbiotic, and that alters the way we view individuals and species. And symbionts can produce new traits in hosts, and that alters the niche of the host. And symbiosis also offers an alternative to the classic neo-Darwinian view of adaptation. Symbiotic adaptation can be rapid. Endophytic fungi were initially thought to be rare, but now we know that endophytic fungi are everywhere. Initially, researchers assumed they were pathogenic. Some of them are pathogenic, but now we appreciate that they have a diversity of function, and some of those functions are context dependent. Many of them are saprotrophs. Endophytic fungi form a holobiont with their host plants, and that affects how we view the concepts of individuals and species and how adaptation occurs. The structure of their communities is determined by the same forces that affect other biological communities, including dispersal limitation, climate, and biological filtering. And finally, the invasiveness of inoculant endophytic fungi and their context dependency may make the use of endophytic fungi in agriculture somewhat risky. Okay, that's it. Thank you very much for your time. I really appreciate it. Turn the time back over to you guys. Let's see.
Okay. Thank you, Prof. Koide. It's nice, really nice and interesting presentation. And we have already, we have already knew the new information from you about the symbiont swapping and the fungal endophytic. And now times to discussion. There are some questions from the participant. can you Can you read?
Yeah, I can see that in the chat.
Yeah.
And
Okay.
I'll try to, is the first one at 7?
From Sophia Lena, yes.
Yeah, Sophia Lena, yeah, okay.
Yeah.
She says, endophytic fungi, oh, very good, have a lot of benefits, but on another hand, some also can become pathogenic to the host. How to manage those fungi to become most benefit to our plants? Second question, since many of the endophytic fungi are very much beneficial, if during plantations we invest endophytic fungi to the rhizosphere, yeah, okay, two very good questions. So first of all, I personally don't have any experience trying to inoculate fungi in the field. We've done a lot of that in the laboratory and in the greenhouse, but we haven't done it in the field. So I don't really have any firsthand experience. Also, I'm not a plant pathologist, so I don't really know, I'm not familiar with all the mechanisms of pathogenesis and, what triggers pathogenesis, when something goes from a latent pathogen to a full-blown pathogen. I'm not exactly sure what happens there. All I can tell you as an ecologist is that things are context dependent, and we can't always predict what triggers the change. So it is always going to be somewhat risky. Let's see. Using rhizosphere endophytes or root endophytes is certainly safer than broadcasting spores across the field. Nevertheless, if you put spores of a particular fungus or some other propagule in the soil, they are going to be in that soil. And if they happen to be not the best fungi for the next crop or the next crop, then there is some danger there as well. Yeah, so I'm sorry I couldn't answer your question any better than that, but.
Okay, and the second one, please, yes.
Yeah, so this is similar to one of the questions previously. I don't really know myself what factors. I think probably some of the plant pathologists in our audience can probably answer this question better than I can. So I don't want to, I probably better not try that one. Sorry about that.
Okay, thank you. And the third one, please.
If we get isolates endophytic fungi from exploration, how to detect isolates endophytic fungi as pathogen or no? Yeah, that's a very good question. So, I'll tell you one thing. It's not good enough to know the species because, for example, with saprotrophy, can they eat litter? There's more variation among isolates within a species than there is among species. And that just means the species name doesn't give us any information with respect to the function. So I don't think you can tell just by knowing the species. I don't know. I don't know how to do that except by trial and error. There may be, when we learn more about the molecular biology of these organisms, we may be able to find certain markers that are, certain genetic markers that can tell us about virulence, about, whether they're going to be bad or good. But right now, I don't think we're sophisticated enough to have any idea. Probably, again, a real good plant pathologist can maybe extrapolate from their experience with known pathogenic fungi. There probably are genetic markers, but I couldn't tell you. Sorry.
Okay. Yeah, okay, it's good. And the next question, please. Yeah.
Since fungal endophytes are fungi, what the effect of fungicide application? Oh, okay. Very good question. So we actually addressed this question in a paper we published maybe 5 or 6 years ago. And we were looking at, in America, many of the seeds that we plant, soybean seeds or maize seeds, cotton seeds, these are all coated with pesticides, including several fungicides. And so we looked at uncoated and coated seeds of corn, maize, to determine the impact that they had on the structure of the endophytic fungal communities. And they had a significant impact on the structure. So, so probably it's these systemic fungicides, as you suspect, that are having an impact on the structure of the fungal communities. And another clue is the fact that these non-mycorrhizal plant species like the Amaranthus or the Amaranthaceae and the Brassicaceae, those species have very strange endophytic fungal communities. And that's possibly because for the same reason that they are not mycorrhizal, not capable of forming symbioses with mycorrhizal fungi. They have some antifungal activity in them that makes it difficult for certain endophytic fungi to gain a foothold in the leaves.
Okay. And the next question, please. From Sriwidi Nugraheni, does the change from endophytic to pathogenic in certain circumstances permanent and includes the change of the genome?
I doubt it. I doubt it changes the genome. Certain things, certain genes are turned on and certain genes are turned off for sure, but I don't think the structure of the genome is going to change. It's just probably gene regulation that's occurring to cause this change.
Okay, and the next one from Muhammad Junay. Simple question is that we have some projects of understanding association of food crops and endophytes undertaken in the nearly future. Do you think we are able to work you and your lab together?
Oh, thank you so much for that. I appreciate that. Actually, I'm an old man and I'm going to retire in 1 year.
Okay.
So I'm not going to start any big new project now. Sorry, but thank you very much.
Yeah. Okay. Thank you, Prof. Koide. And the other question from Liania Sartika. Based on your explanation, as I get that microbes has their own community in a group. So it means that the controlling system to the plants are also can be considering by the what kind of microbes are in the community. Do you think if we isolated 1 or 2 microbes from community, then commercialize it, it would be worth?
Yeah, that's a very good question. So as I showed in the data earlier, different plant species harbor different communities. That means it's not always that they are completely different fungi, but certainly different. The distribution of the abundance is different. And in some cases, there are certainly some fungi that can colonize some plant species and cannot colonize another plant species. So commercialization of an endophytic, a beneficial endophytic fungus is probably going to have to be specific to a particular crop species. So if you wanted to commercialize one for maize or one for soybean or one for this or that, okay, but probably not one for all of the species, probably something like that.
Okay, thank you, Prof. Koide. Is there any question from the participant, please? We still have a lot of time. Okay, from me, maybe Prof. Koide. How can endophytic fungi break down the cellulose of the plant part? Have you already searched from the endophytic something like the secondary metabolites produced by the endophytic the fungi?
Yeah, we don't know anything about the biochemistry of saprotrophy, but I suspect we did all of our tests with oak leaves, and oak leaves obviously contain cellulose and lignin, but also a very high concentration of polyphenolic compounds like tannins. So I suspect that all of these fungi, or at least most of them, can eat cellulose quite easily. But the difference in their saprotrophic ability probably has to do with their ability to tolerate the polyphenolic compounds. Not all fungi can tolerate the polyphenolic compounds very well. So, but I don't know anything about the enzymes or anything like that, although I suspect they're all pretty good with cellulose.
Yeah, right. Because a lot of endophytic fungi can break down the, for example, the chitin or the cellulose, something like that. Yes, there are another question from Abdul Ghafur. In certain case, people compare the nature of interaction between host plants and endophytes and interaction between host plant and growth-promoting microbes. Would you comment on it?
Yeah, like plant growth promoting rhizobacteria and that sort of thing. I don't know the literature very well. I think some of the growth promoting rhizobacteria are producing plant hormones of one kind or another, although I'm not certain of that. I don't know of any reports of endophytic fungi producing plant hormones, but they could certainly stimulate the plant to produce hormones. I showed you one photograph of an endophytic fungus that does something similar to what other fungi do. It looks like it's stimulating cytokine production or making cytokines because it has those little green islands, you know, in the leaves as the leaf senesces. So there could be some, yeah, there is a comparison that could be drawn. Probably there are some effects of the endophytic fungi on plant hormone production, or they may produce plant hormones themselves. I'm not sure.
Okay. Is there any question from the participants? We still have enough time. So another question from me. What kind of factor influence to the microbes become endophytic fungi to establish in the plant?
Can you say that one more time, please?
Yeah. What kind of factor influence to the certain microbes become endophytic microbes in the plant, can establish in the plant?
What plant factors?
Yeah, what kind of factors? So the endophytic can become established in the plant.
Yeah, that's a really good question. And I think it depends. So, you know, it's like everything else. It depends. So the kind of endophytic fungi in cool season grasses that grows throughout the length of the leaf, those are very, very different from the class 3 endophytes that we have studied primarily. The class 3 endophytes are probably not very good biotrophs. They don't grow very extensively inside the leaf. And I'm not sure if that's because they're not biotrophic or because the plant is actually putting up some defenses. I don't know. But the other ones that produce the alkaloids, they're growing extensively inside that leaf. So either they do not stimulate plant defenses, or if there are plant defenses, they have some way of counteracting those defenses. I'm not sure which, but different kinds of endophytic fungi probably colonize the tissues through different mechanisms.
Okay, thank you, Prof. Koite. And there's another question from Prof. Suganda, Prof. Tarkus. I just want to make sure, is there any single plant that has no endophyte associated with it?
Well, that's impossible to answer because people actually haven't examined all the plants in the world. But I can say that of the few plants that have been examined, hundreds of them, nobody has found a plant that was not colonized. Some are colonized very heavily, some are colonized very poorly, but it seems like all of them so far have been colonized. Now, we may find some surprises. Initially, You know, mycorrhizal fungi were thought to colonize all plants, but now we know that there are a number of plant species that are not colonized by mycorrhizal fungi. So it's possible. But I'll say this, endophytic fungi are not a single taxon. It's not a single group of fungi. It's all kinds of different fungi. Most of them are ascomycetes. Some of them are mucormycetes. Some of them are basidiomycetes. So they're all over the map. And so it's, if a plant has a defense mechanism against some of them, I doubt that it would have a defense mechanism against all of them. So it's, I think in my mind, it's not likely to, we're not likely to find a plant that's not colonized by some endophyte.
Okay. And another question, is there any potential fungi to degrade liquid waste?
Who can degrade liquid waste?
Yeah, that one.
Yeah, probably. I mean, it depends on the nature of the waste, but fungi make some pretty sophisticated enzymes. So I think when it comes to hydrolyzing or oxidizing or this kind of activity, if you could make a generalization, it would be that fungi are more competent than bacteria in doing this. There are obviously exceptions, but as a rule, fungi can make some pretty sophisticated enzymes to break down very, very complex materials. So I would guess there are some fungi somewhere out there. The best place to isolate those kinds of fungi would be in the liquid waste itself to see if something's growing in there.
Okay. And another question from Liania Sartika. I'm working on Pseudomonas chlorophragis that is isolated from banana plant. Using molecular detection by designing specific primer of this species, I could detect the bacteria after 3 years again. But when I try to isolate it, I'm not able to isolate this bacteria. I may conclude that probably this bacteria growth can be suppressed by other microbes secreted metabolites, but I'm not sure. Do you have an idea of my experience?
Yeah, that's a good hypothesis. There's another hypothesis, and that, you know, my experience is mostly with soil, and we know that we can isolate bacterial and fungal DNA from soil that is just free DNA, and it's just adsorbed onto soil particles, and it's not inside a living organism at all. So it's possible that when you isolate from bananas, you're getting the DNA, but it's not coming from a living organism. It's just naked DNA that's somehow preserved on a surface somewhere. But your hypothesis is also good. It could be that there are something that suppresses the growth of the bacteria.
Okay. And another question from Alfi Inayati. If we want to isolate endophyte fungi, which part of the plant is the best to get abundant fungi?
So the communities of the endophytic fungi are going to differ in different plant parts. So it depends on what you want. The communities in the leaves will differ from the communities in the stems, and those will differ from the communities in the roots. And they will even differ in different kinds of roots. So it's, and in the flower will be different. So every plant part will be different. The easiest kinds of tissues to isolate fungi from are those that can tolerate surface sterilization. So when we do this, we dip the tissue in ethanol, then we dip it in bleach, then we dip it in ethanol, and then we let it dry, and then we put it on the agar. So if the tissue is so soft, like maybe petals, maybe that wouldn't be able to tolerate that kind of surface sterilization. But unless you kill the DNA and the fungi on the outside, you don't know that you're getting endophytes. You could just be getting epiphytes, surface fungi.
Okay, good. Thank you. And the other question from Dr. Muslim. Another benefit of endophyte fungi are they act as biocontrol agents, namely within the mechanism of competitive with pathogen and induced resistance. How about your opinions?
See, biocontrol agents namely within the mechanism competitive with pathogen and induced resistance. Induced resistance. This is possible. I don't know of any research suggesting that these work that way, but I don't see why it couldn't. Because again, endophytic fungi are not a discrete group. They have no single function. They are not a single lineage of fungi. It's just anything that can grow inside the fungus. So anything a pathogen can do, anything a mutualist can do, anything any other fungus can do, probably you're going to find among this group of fungi because they're so variable. So I'm sorry, I can't answer that question very well at all.
And another question from Hardian Susilo Adi, Jember University. Could you tell me what is the most interesting benefits of endophytic plant interaction that probably has a great prospect in the future research, such as the utilizing endophytic for supporting the growth of plants?
Yeah. Well, again, this is we have to look at the context here. In your country, maybe the benefits you want to derive from endophytic fungi are different from the ones we want to derive in my country, or the west coast of the United States and the east coast of the United States. Very, very different climates. So if you're in a place like Utah or the west coast of the United States, then drought and heat are very important stresses we have to deal with. But if you're where you are, maybe heat is a difficulty, but probably not water stress so much. I don't know. So I think it depends on the situation. But we have found that some fungi are good with resisting insect herbivory. Some are good at increasing resistance to drought or heat or even cold, and so forth. So I think it depends on the particular situation. Again, there's not one fungus that's going to be good for every situation or for every plant species.
Okay, thank you. And the other question from Mutalalia. I would like to ask whether the use of endophytic fungi to control the same disease can directly apply in all locations, or it must be specific location.
Yeah. Well, first of all, I don't know. I don't know of any research that shows that endophytic fungi can control any particular disease. Although that's possible, I'm not sure of any research on that topic, so I really can't address that. But on general principle, I would guess disease, as you know, is context dependent. Disease depends on, you've got to have the disease organism, but you've got to have the environmental conditions that precipitate disease. So, some diseases occur when it's cold and wet. Some diseases occur when it's hot and wet. So it all depends, I guess, is the answer.
Okay. And from Ratnawati, if isolate endophytic fungi that sometimes different growth, early isolate endophytic. What's the fact? So she tried to isolate the endophytic fungi, but the other time has the different growth isolate.
Yeah. Well, as I said earlier, we found that there's a lot of variability among isolates of the same species. So even if you isolate the same species, if you isolate the same species 10 times, they will all be different, 10 of them different functions. So it's very, very difficult. And for all of you know who've grown fungi in culture, you know that a single isolate can change in the culture. So if you culture it for more than a few weeks, you can get a somatic mutation or something. And it's just different. Not only it looks different, but it acts different. So these fungi are not stable. It's difficult. Yeah.
Yeah. And from Haryadi, we have some mechanism to attenuate the virulence of pathogen bacteria. For example, quorum sequencing mechanism. Is this mechanism can be happen on endophytic fungi too?
Okay, that's a question that's way above my level of understanding. I don't understand what quorum quenching is. And so I really can't answer that question. I'm so sorry.
Okay.
You got me on that one.
Yeah. Okay, thank you. And from Ani Widya, Ibu Ani Widya, based on your research, do you think that interaction endophytic plant that help plant growth needs fungal or microbes in community? Do you think to isolate one or some species of endophytic fungi and treat or give to the plant will not fully work as they work as community?
Yeah, that is a very good question. In nature, in these class 3 endophytic fungi, we never find a plant that has only one fungal species in it. It's always, maybe dozens or maybe even 100 different species of fungi in one leaf. So I don't know, even if you were able to find one isolate of fungus that was beneficial and you inoculated that, it's not going to stay one species. Over time, it's going to be colonized by many things blowing around in the air. And so it doesn't happen in a field that you can keep one species in it. So I think it's going to be very difficult to test whether that is true or not.
Okay, thank you. Is there any question from the participants? We still have more or less 5 minutes. The time. From Dr. Abdul Ghafur, do you expect immediate positive contribution for endophytes to other vital aspects, for instance, human health?
That's a good question. We have already seen that some endophytic fungi can produce like Taxol and other medicinal compounds. In fact, there's quite a bit of research being done in the Middle East because a number of scientists think that a number of these secondary metabolites that we thought were produced by plants that have beneficial effects on human health, they are actually produced by the fungi inside the plants and not the plants themselves, or at least some interaction between the fungi and the plants. So yeah, that's a really good question. I think we probably can expect to find that endophytic fungi will contribute to even to human health.
Yeah. Okay. One question again from the participant. If there is a question before I close. No? Okay, Prof. Koide, is really nice information, right? Very nice science from you. And thank you so much for your share, for
Thank
your
you.
event. And I really appreciate for you. And as our appreciation, please the committee share a certificate and drawing from local artists of East Java province for Professor Koide.
Thank you very much. I appreciate it. Bye-bye.
Yeah. Wait a moment, Professor Koide.
Okay.
We share a certificate for you
Oh.
and a drawing. Yeah. Please, the committee.
Professor Lucas, excuse me to interrupt your moderating. For the certificate, we will send to Prof. Koide right after the international conference, but now we would like to show you the result of graphic recording during the session of Professor Koide. And this is the graphic recording.
Yeah. Okay. Thank you. And from knowledge and sharing the science from Prof. Koide, I should conclude with two things. That the first, the symbiont swapping can lead the rapid adaptation. And second one, some endophytic fungi have many advantages and functions. And that's the time with Professor Koide. And thank you so much, Professor Koide. And I hope you are always in good health and success for your future. Thank you, Prof. Koide.
Thank you very much.
Yeah. Okay. Now we come to the second speaker. Yeah. To the second keynote speaker, we have Professor Wei Jiangshan from National Taiwan University. Please, the committee can share the Professor Shan curriculum vitae.
Can you see my screen?
Professor Wei Jiangshan got his PhD from Texas University on plant pathology and microbiology. And his research and experience, a lot of experiences. And he became a president for Mycological Society of Taiwan and become the head of Department of Plant Pathology and Microbiology, National Taiwan University, and become a professor 2011 until now from Department of Plant Pathology and Microbiology, National Taiwan University. And he has a lot of experience about the plant disease. And now I give the time and screen for you, Professor Shan.
Okay, thank you very much. And can everybody hear me?
Yes,
okay.
We can hear you.
Okay, thank you.
Okay. First of all, I would like to thank the Indonesia Phytopathological Society to invite me, and also colleague from University of Brawijaya to invite me to join this conference. And so, okay. And also, I also would like to thank the moderator to introduce me. Okay. It's my honor and privilege to present our work in this conference. Okay. And can you guys see my PPT? Yes. Okay. Wonderful. Okay. So today I'm gonna share you about our work in rice blast population study. So the title is Surveillance of Dynamic Rice Blast Field Population Fungus population in Taiwan. And first of all, I would like to dedicate this work to Professor Lily. Okay, I think all of you know Professor Lily well. Unfortunately, she passed away a few months ago, and so I hope she is in peace, and also Hello, Professor Sam. Is there any trouble? Okay, now please continue, Professor Sam.
Yeah, Pak.
Okay.
Okay, sorry for the internet issue. Okay, and
No problem.
it's okay, right? Okay, and I also would like to take this opportunity to acknowledge two of my students. So the first one is Jaha Saoki. Oki is a double degree master's student from University of Brawijaya, and so Professor Lidis established this double degree program between University of Brawijaya and National Taiwan University, and so Oki is a very talented student and smart. He is very smart and also diligent, and so he spent a lot of time for the work. Okay, and another student is Bore, and so Bore is from Taiwan. So two of the students involved in this project. Okay, but I think this is just brief background. Since since the moderator already introduced, so I got my PhD from Texas A&M University. Okay, and I was the chairman for the department and National Taiwan University a few years ago. I'm currently is the president of Mycological Society of Taiwan. Okay, and very brief. So my research interest is plant pathology and also fungal biology and genetics. So in my laboratory, I work on mainly on fungi, and especially on the plant pathogenic fungi. And so we work on rice blast fungus. Today I'm gonna introduce our work. And in our lab, we also work on Panama disease, Fusarium oxysporum forma specialist cuban. And we also work on another smart fungus. It's called water bamboo smart fungus. It's an edible vegetable due to the infection of this smart fungus. So we studied the molecular mechanism of tissue infection and growth formation of this smart fungus. So this is the three main projects related to fungi in the lab. And recently we are working on the biocontrol of Panama disease, and also several students from University of Brawijaya. And I think the first one isolate this bacteria is Yosa. Yosa also from University of Brawijaya, and I believe he's on the audience. So he isolated a lot of bacteria and fungi from the rhizosphere of banana root. So one of the bacteria he and other student work on is called Pseudomonas chloranthes. So this bacteria show the PGPR activity and also antagonistic activity for FOC, and so this is another project in the lab. So very brief to introduce the research works in my lab. Okay, so if anyone from the audience or from your society interested in these topics, you can contact me later. Okay. Okay. So let's back to today's focus. So today I'm gonna share our work from on the rice blast disease. Okay. As you can see, so rice blast is very important disease of cultivated rice around the world. So it can cause leaf blast. And so the photo on the screen, you can see is a typical lesion of rice blast disease. And at the same time, rice blast fungus can cause panicoblasts. So if panicoblasts occur, that will severely decrease the yield for the farmer. So at the lower right hand side is a screenshot from the news. So in Taiwan, once in a while, we will have the panicoblasts that can reduce the yield maybe around 30% to the farmer. Okay, so this is rice blast, but I think you all know and the disease triangle concept in the plant pathology field. So in rice blast, the host has susceptible and resistant host. For the pathogen, rice blast fungus has a lot of physiological races, and if there's a host and pathogen, for the disease to occur, it also requires the environmental condition, such as temperature, high humidity, and also the soil condition. Okay, and in the photo I show in this slide, you can see in the same field, same variety. This field subjected to the same pathogen population, but you can see on the right hand side there's a disease kind of severe, but on the left hand side the rice is quite healthy. Okay, so I think this may be due to the soil condition in the difference in these two fields. Okay, and to manage this disease, there are several control measures, and of course traditional way is the chemical control. I think for this disease, cultural control is also very important. So, for example, not to use a lot of nitrogen fertilizer. Okay, and an environmental friendly, ecological friendly way to manage this disease is to grow the blast resistant variety. But in different country or different rice growing region, people use rice blast resistant variety. However, the population, pathogen population is in a field are quite dynamic. So rice blast fungus is has a lot of mechanism to become virulent pathogen. So to use this blast resistant variety effectively, that require disease monitoring, especially for the pathogen population. Okay. This is the life cycle of rice blast fungus. In the most of the rice cultivated field only have sexual reproduction cycle of this fungus. So in a commercial field, the major propagator for the disease is the Canidia. Okay, so the Canidia will infect the area part of plant, and rice blast fungus belong to Ascomyces, and it has typical parasitium ascus and ascuspore structure in Ascomyces. It has two mating type 1-1 and mating type 1-2. And so mating mating is not normally occur in a commercial rice field. However, the mating may occur in some specific region, like the area near the origin of rice, like the northern part or southern part of Himalaya mountains. And so in those areas, maybe there's a sexual reproduction in the natural condition or field. Okay, and I would like to focus on the disease cycle. So rice blast fungus has a lot of weapon for attachment, and also it will produce this specialized turgor pressure generated infection structure called Aposorian. And once rice fungus penetrate to the rice leaf, a lot of interaction occur. And after penetration, the rice blast fungus can secrete a lot of effectors. There are two major types of effector. The first type is the cytoplasmic effector. This type of effectors secreted through a specialized highly membrane-bound complex called biotrophic interfacial complex, the BIC. And that BIC will secrete the cytoplasmic effector from the fungus to the rice cytoplasm. And there's a second type of effector called apoplastic effector. And rice blast fungus can also secrete this type of effectors through traditional secretion pathway. And so, there are two types of effector function after penetration. This effector will manipulate host physiology, metabolism, and immunity, and to help the fungus to successfully infect the plant. Okay. And this is just one summary slide for the work done by initially done by the Professor Barbara Valen in the United States. And so by finding a lot of effectors from the rice blast genome, and they conduct the very elegant fluorescence study by labeling different type of effector with fluorescent protein. So as you can see, there are effector, cytoplasmic effector label in red, red fluorescent protein, and it can secrete through big and localize into the rice cytoplasm. In this case, they label this, they engineer this effector, so the effector, fluorescent effector, can be seen clearly in the rice nucleus. Okay, so you can see the infected cell, where infected cell has this red signal, but the surrounding rice cell also have this effector labeling in the nucleus. But the second type of effector is apoplastic effector. That effector labeled in green. So it outlines the invasive hyphae for the first invaded rice cell. Okay, so these effector are quite a lot of research focused on this effector for the past 10 or 15 years. Okay. Rice and rice blast fungus is a model system, one of the model system to study plant and microbe interaction. Okay, so rice blast fungus genome is sequenced. So based on the prediction, there are a lot of secreted protein, maybe up to 1,500 secreted protein in the rice blast genome. And there are more than 16 effector protein has been well categorized, including 10 evidence effectors. And okay, and on the other side, the host has a lot of resistant gene has been categorized, has been identified or cloned from different rice variety around the world. Okay, and among those blast resistant gene, there are over 20 are well categorized at a molecular level. Okay. This is the old model, the gene for gene concept in plant pathology. In rice and rice blast system, the vertical resistance is, there are quite a lot of examples are well studied. So in this gene for gene model, if for the pathogen, if the pathogen contains the avians genes, and that avian gene is recognized by the corresponding resistant gene in the host, that will trigger the incompatible reaction. Or nowadays people know that is a ETI, the effector trigger immunity response. Okay, but however, if the pathogen avian gene got mutated, or the population of pathogen do not have this avian genes, so it can infect. They all can infect the resistant rice variety. So this is the gene for gene concept. In the rice and rice blast system, so the top one, so if the pathogen has avian gene, then can be recognized by the corresponding resistant gene in the rice variety. And that the resistant or incompatible response will induce. So the rice plant will have good resistance against this population. So the rice, the farmer will have the rice harvest. So this is kind of the breeding purpose, to breed for the rice variety with effective resistant gene. However, if the population, the corresponding, the AVR gene got mutation and no longer recognized by this R gene, this population will break down this rice variety. Okay, so this is the vertical resistance in rice. Okay, sorry. I would like to use the one old and well-studied example is the PTR gene from rice and then AVR pita, avian gene from the rice blast fungus. And so the top photo show you the 4 conditions of this gene for gene concept. The number 3 condition is the rice variety containing the pita resistant gene, and the infected pathogen has corresponding AVR pita avian gene. So the plant is healthy and grow better than the other 4 conditions. And so if show the leaf from this, the material containing the pita, so there's no lesion develop from this kind of reaction. In the other case, then the other 3 cases, the typical lesion will form on the leaf of those rice interaction. Okay, so AVR pita initially identified as a telomere of chromosome by map-based cloning. Okay, so it's a telomeric avian gene, and that will determine the efficacy of the pita blast resistant gene. But from the population study of the pathogen, a lot of pathogen has mutation of this AVR pita genes, including the point mutation, insertion, deletion. And this kind of mutated isolate and can infect the rice variety containing pita resistant gene. Okay, as I mentioned. This is one example study in the United States. So, and they collect the isolate from the pita resistant variety, and they found in AVR pita gene among those virulence isolate, there's a insertion occur by the PO3 transposon. So PO3 is a transposon in the rice blast genome. So this PO3 transposition in this isolate will disrupt the AVR pita function and cause the convert the avian phenotype to virulence phenotype of this pathogen on the pita resistant variety. Okay, this is one example. Okay. Sorry. So in traditionally, so to know the population, pathogen population structure in the field is very important for management, okay, or to deploy the effective blast-resistant variety. Okay, traditionally, rice blast pathogen or can be differentiated into different physiological races by rice differential cultivar. Okay, so the photo show you is the international rice differential cultivar. There are 5 different rice cultivar from different countries, including the Philippines, United States, and China, and India, and Pakistan, and also Japan. But however, this system is old, and the variety for those cultivar, the resistant gene content in this cultivar are not well categorized. So in this case, it's hard to use this information for breeding and also to differentiate the race efficiently. Okay, this is the old system. But this table shows you that there are a lot of blast-resistant gene identify, clone, and categorize, including the one I mentioned, PITA, and also other resistant gene like PIZT resistant gene and PIK resistant gene. Okay, and because this the gene or blast-resistant gene or locus has been identified from different rice variety or cultivar. So the researchers in the Erie International Rice Research Institute, they collaborate with Japanese scientists. They developed the IRBL lines for rice blast disease monitoring. So they use different rice variety or cultivar with identify blast-resistant gene. They use that as a donor to integrate into susceptible rice variety. In this case, it's LTH. LTH is a indica susceptible variety from China, southwest of China, the local variety. So that variety is susceptible. So they use the traditional breeding and molecular marker to confirm the breeding material containing individual blast-resistant gene or locus. So in this case, totally there are 30 monogenic line they develop in this LTH background. So as you can see, these different IRBL line, they have different rice blast-resistant gene or locus. Okay, so one case here is I highlight in red is that's a PIK resistant gene. This PIK has different allele reported from different rice variety. So they integrate different allele into LTH. So some allele is resistant allele, and some allele may not resistant to the certain race of the rice blast fungus. Okay, so this IRBO line is very useful to monitor the effectiveness of the blast resistant gene. Also, very useful to monitor the virulent pathogen population in the field. Okay, so some years ago, the researcher we collaborate with the IRI, so we introduced this system into Taiwan. So we grow this IRBO line in Taiwan in 6 different locations. In the southwest part of Taiwan is the main rice production area, including Pingtung, Tainan, and Jiayi. This area is the major rice production area. And we also grow this material in the eastern part of Taiwan, Taidong, and middle part, Nantou, and northern part of Taiwan, Taoyuan, to monitor the effectiveness of blast resistant gene and also population in the field. Okay, so very brief to show you the upper photo is the plot set up in Taidong, and in Taidong, and as you can see, the IRBO line containing blast resistant PIA and PII. The number is 1, 2, 3. So these 3 lines has legion we collect from the field as shown in this here. Okay, and on the other hand, and there are line 22, it containing PI9 resistant gene. Then line 27 and 28, it containing PTA2 resistant gene. And this PTA2 recently cloned and renamed as a PTR resistant gene. Okay, so PI9 and PTR, these 3 varieties show very good resistance. They are quite healthy in the field. Okay, so this is the monitor of the we monitor the disease with this material in the field. Okay, and this here is another example. So again, PIA, PII is not effective in the middle part of Taiwan, in Nantou. But PTA2, the PTR, is still very consistently effective against the population in Taiwan. Okay. Okay. So this is the R gene status for our monitor. So for the following of my presentation, I'm gonna focus on 3 systems. Okay. And the first is PIZ-T and AVR-PIZ-T. And also the second one is PIB and AVR-PIB. Then the third one is PIK and AVR-PIK. These 3 pairs of the R gene and AVR gene of our study. Okay. And so over the years, each crop season, we collect a lot of rice blast isolate in order to separate or selected rice plus representative isolate for our study. We first use fingerprinting techniques to kind of categorize the isolate we collect. So we use PO2 red PCR fingerprinting technique to study the population. So as you can see, we did single spore isolation. We culture the fungus. So we have a lot of culture. So we extract DNA. We conduct the PO2 red PCR. So on the left-hand side, this is the we run the PCR result on the gel, and you can see this is the sample we collect from Tainan, and from the year 2017. It can kind of separate into two major lineage, two major groups, okay? The G1 and G2 group. So G1 group is quite, the pattern is very similar to each other. Then G2 group also very similar to each other. But G1, G2, the pattern, we can differentiate by this PO2 red PCR fingerprinting techniques, okay?
And Professor Sen,
but to brief, PO2 is also the retrotransposon in the rice plus fungus genome. There are more than 100 copies in the genome. So Professor He Liang also in Erie, he developed this technique quite a while ago. So we can design primer in a PO2 conserved sequence. So this red PCR will amplify the intergenic region among two copies of PO2. So this PCR fragment amplify around the gel, so we can differentiate. This is fingerprinting amplification we can use to dissect the lineage of population from the field. Okay. So the first example.
Hello, excuse me.
So the first example is the AVR PIZT resistant gene. So this is the sample, the isolate we collect from Pingdong, and as you can see, by running the AVR, the PCR product with the primer of AVR PIZT gene, the lower band is the wild type copy. The upper band among this isolate is about 2.2 kb. So you can see only these two types of the amplification pattern. So we further sequence and we find this AVR PIZT resistant gene. There's a PO2 insertion, like the example I show you in the AVR PETA case studied in the United States. So there's this dominant one, okay?
Excuse me, Professor.
Okay, so this is the diagram show you. There's a PO2 insertion inside the open reading frame of the AVR PIZT gene. So the insertion will cause the disruption of the AVR PIZT function. So the isolate will be from the areas become virus. Okay. Okay.
Excuse me, Professor. The time is.
And this is the result of combine the PO2 fingerprinting and AVR PIZT resistant gene. So based on PO2, we can separate into 2 or 3 different groups. But you can see in the G2 group, all the AVR-PIZT resistant AVR gene are got PO2 insertion that's mutated. But the other, the G1 group, the G1 group, the AVR-PIZT gene is intact, maintain the wild type avians function. So, so this AVR-PIZT genotype kind of correlate with our fingerprinting result. Okay, and just another quick information. We also tried develop the face detection protocol. Use the portable machine and use the rice blast legion, and we did a quick extraction. Then use the like qPCR detection rationale, and use this portable machine. We can quickly detect the AVR-PIZT lesion status in the field. Okay, this is the qPCR Tickman system. Okay, on the screen we can see the signal, and okay, this is the face detection tool we developed for this gene. The second system is the AVR PIB gene polymorphism study. Okay, and PIB plus resistant gene is quite resistant in different locations of Taiwan over the years. But however, in recent years, we notice in few locations this IRBO line show lesion in few locations. So as you can see, this is in 2017, 18, 19. So in the early, like 2017, in the same location, like Nantou, the plant is healthy. But in recent years, there's a few lesions occur, okay? And this is the PIB resistant gene. So, so over the year, we collect, especially you can notice in the table, in the year 2019 and 20, we have a lot of isolate from Hainan, Pingtung, and Taitung. Okay, so this AVR PIB gene is cloned, so we can design primer to screen. So you can see the wild type band is smaller, it's about 1 kb, but some isolate we can amplify bigger size of the product. So we design different primer can amplify different part of region of the AVR PIB locus. So so we can identify where is the insertion occur among those virulence isolate. So after all this study, the screening and the sequencing of the PCR product, and we can identify 8 different virulent genotype. Okay, so majority of the virulence isolate, they have PO3 insertion in the promoter or the operating frame. Okay, then there are also other type of mutation. For example, there's an MG site inserted in the promoter region, and there's a one type is lack of the AVR PIB gene in their genome, and the type 4 V type 4, they are double allele. So this double allele is a novel finding in our study, has not been reported in the literature. Okay, this is the AVR PIB. Sorry. So we performed the inoculation. Okay, I think this inoculation done by Orchid. So you can see the original LTH susceptible variety or the variety containing PIB resistant gene. This is the wild type. So wild type strain has intact AVR PIB, cannot infect PIB line. However, the other virus genotype, since the AVR PIB gene got mutated, they all can infect this PIB line. Okay, as demonstrated in this inoculation result. Okay. So we also study the virus isolate distribution, this virulent type distribution in Taiwan. Okay, and so I think a lot of virus isolate occur in Tainan and also in Taidong area. But this we collect this isolate because we use the PIB, the IRBO line with the PIB resistant gene. So that plant kind of select the virus isolate, allow them to grow, infect this variety in the field. But the real population in the field is not that, the proportion of that virus isolate is not that high, as demonstrated in the right-hand side. We also use the lesion, single lesion quick detection protocol in Pingdong. So only 1 out of 72 lesion is a virus isolate. So in the real population, the virus isolate is not that high in the population. Okay.
Excuse me,
but we also kind of put the virus isolate, the genotype on top of the PO2 phylogenetic tree. So we can see this mutation occur probably both in G1 and G2. Okay. So that indicate this recent mutation event of AVR PIB gene probably occur independently in the population. Okay. Okay. I think time is kind of up. So, but let me quickly within 3 minutes, I will finish my talk and for discussion. So I think I use PIZT and AVR PIZT and AVR PIB as an example to show you our study. But we also conduct other AVR gene screening. I think there's one AVR gene is very complicated. It's called AVR PIK. Okay, this AVR PIK gene has more than 5, and right now probably over 6 allele, including A, B, C, D, E allele. Okay, of AVR PIK genes. Okay, and this AVR PIK and PIK gene has been the subject for unenforced co-evolution study from other laboratories, and so R gene evolved, and AVR gene also evolved. You know, so this is the idea of unenforced. You know, so in this case, the AVR PIK allele, D allele is the dominant avians allele, can be recognized by different PIK resistant allele, but cannot be recognized by the susceptible allele. And this is the D allele. Then A allele is also can be recognized predominantly by the PIKM allele of PIK gene, okay, but not recognized by other resistant or susceptible allele. So this is kind of system in the AVR PIK and PIK. So it's kind of complicated system because in this system, it's not at a gene for gene level. It's function at a allele to the allele specificity level, okay? But again, we also screen this AVR PIK gene status, like by designing the primer or the specific primer for A and D allele, okay? Because in Taiwan, there are two dominant allele, A and D allele among population. But however, there are the GIF picture show here, there are at least probably 9 or more than 9 different genotype of AVR PIK allele among Taiwan population. So the lower part is the wild type copy of open reading frame. The length one is the AVR PIK A allele amplification. Length 2 is D allele amplification. So A and D is two dominant allele, two allele present in the genome. Okay, but as you can see, there are different kind of combination of this AVR PIK genotype among population. Again, we also did an inoculation. So if there's an A and D allele or functional D allele, but so those variety has functional PIK resistant gene, this avians isolate cannot infect. Okay, but however, if in other type of mutation occur, it can infect this PIK resistant variety. But this is again fit to the PO2, so it's a result. Okay. Okay. We also monitor the population in different locations over the year, and so this is the result. Okay. So the composition or dynamic in different locations over the year are quite dynamic. Okay. So if we use these 3 AVR genotype, we can differentiate 21 genotypic group of rice blast population in Taiwan, 21. Okay, this is the group in different locations. And if based on the infectivity of this isolate on PIZT, PIK, and PIB rice variety, okay, we can differentiate the pathogen into 8 different races, okay, as shown here. Okay, so this is the distribution of race 1, race 2, race 3, race 4, race 5, okay, in different location in Taiwan. Okay, so this is the overall result in different location in terms of race distribution. Okay, in summary, so this is the work we conduct. This is a team effort project. We grow this IRBL line in the field, so we can use this line to monitor the R gene effectiveness. And at the same time, we can use this material to monitor the virulence isolate in different locations in Taiwan. But interestingly, different AVR gene subjected to different type of mutation. So for the moment, we don't know how exactly that occur for those 3 different AVR genes, you know. But I think our surveillance study provide very useful information for R gene status, or maybe in Taiwan, and also this effective R gene can be used for breeding and deployment of resistant variety. Okay. Sorry, I overtime. And finally, this work is a team effort project. A lot of researchers in Taiwan from experimental station help us to grow and to sample the material from the field for us. Okay. Thanks for your attention, and I will take.
Okay, thank you, Professor Sen, for your nice presentation.
Thank you.
And now we still have only 15 minutes to come to the discussion.
Right.
And there are some questions for you. And one question is related to the molecular genetic from Ali Wafa. Can you read the questions in the chat public? Question from Ali Wafa: do you have some methods for induce the plus fungi Viricularia oryzae and push them sexual cycle or entering their telomorph phase? We know in equatorial country like Indonesia, almost impossible to see Magna Porte oryzae in rice environment. And the second one, in this decade, the genome editing tech are developing technique to build and boost some resistant gene in plant, like for the rice RR gene, or just for elicitor and effector mining. In your opinion, how much we needed them and how necessary we must change the traditional breeding to genome editing technology? Please, Professor San.
Okay. Thanks for the question. And for the first question, I think people use the oatmeal agar and to induce sexual reproduction on a plant in a laboratory condition. Uh, 6 degrees Celsius on oatmeal agar. Maybe dark condition is better than light condition. So you can set out a daylight cycle, probably long dark cycle, short light cycle. Okay, so I think for fertile strain, normally we can see this, the sexual structure maybe after 2 weeks incubation. Okay.
Okay.
But so if you want to perform this experiment, it's better you have fertile strain as a control in the lab. Some fertile strain are collected from the non-rice hosts. For example, from other wheats. Okay. because you need to have a control to show the condition works. And we also try to grow to induce sexual reproduction for those rice isolate we collected from the field. But however, so far we cannot see the sexual reproduction cycle structure in the laboratory setting. Okay, but for the case in Taiwan, for the majority of the rice isolate, they are all one mating type. Okay, but recently we identify another mating type from one or two locations. We try to make those strands, but so far we cannot see the sexual structure out of that those strands. So for the first question, I think there's a condition we can induce, but you need to have a fertile strand as a control. Because in the literature, people also report a lot of fear isolate from the rice plant. Those fear isolate has fertility defect. So you can try. Okay, you can try. So this is the first answer for that. Okay.
And for the second one?
For the genome editing for blast or for the rice blast resistant gene, I think this genome editing technology has been well developed. Okay, so there are few rice researchers in Taiwan. They have this kind of tool. Of course, in other countries, there are a lot of researchers using this tool, genome editing tool, to edit rice genes or rice blast resistant gene. Okay. So my suggestion is you can collaborate with the rice researcher for this topic to set up the protocol in your laboratory. Okay. So of course, compared to the traditional breeding, the molecular breeding is quicker, especially for this genome editing, probably quicker. But in Taiwan, GMO is not allowed to grow in a field. So far, the genome editing rice or plant material we are also in the discussion whether we consider that as a GMO we can grow. But a lot of rice breeder in Taiwan, they use molecular marker assist breeding. I think that can help shorten the breeding period compared to the traditional breeding. Okay, so other researchers in this team effort project, for example, Professor Zhong in our department, and he kind of integrate this novel or effective blast resistant gene into Taiwan commercial variety. For example, PTR, PI9, loss blast resistant gene into rice elite commercial variety in Taiwan. Okay, so I think this is the first question. First, the answer for the first and second question from one audience. Okay.
Okay. Thank you very much, Professor Sen. And we still have time. And the second question from Sophia Lena. Yeah. Regarding to your research, how the climatic factors influence the rice blast disease in your place? And the second one, did you do research regarding to gene manipulation to increase plant resistance to rice blast?
Climate.
To answer the first question, of course, climate change affects the rice blast disease. I didn't mention in Taiwan, we grow rice for 2 crop seasons. The crop season, first crop season is the first part of the year, maybe from January to June. The second crop season is from the June or July to November. Okay. Normally, rice blast disease occur in the first crop season, not in the second crop season. Because the temperature, the humidity is not favorable for disease to occur in the second crop season. Okay, but the disease occurrence in the past few years, the pattern is kind of changed. Okay, the disease occurred a little bit earlier in the first crop season. Okay, so I think climate change affect the disease. I would not say probably the disease will reduce due to climate change, because if we have a hotter weather, maybe not that favorable for the disease. But I would say probably the early crop season of the first crop season, the early period, or the late period of the second crop season, maybe the rice blast disease, either leaf blast or panicle blast in the second crop season, maybe will occur. So definitely the climate change affect the pattern of blast disease in Taiwan. Okay, this is the first question.
Okay.
And for the second question, I'm not sure the audience asks the gene manipulation for the blast resistant gene. If that's the case, I personally don't have time or or people to work on rice plants, that part, whole side. I mainly focus on the pathogen side, but the colleague in my department or other institute, they focus on that part from the host sites. Okay.
So, okay, I think it's clear that your answer, and I hope the question is already answered well. And I do apologize because of the time. We do not enough time to continue deliver the question from the participant. So if you have time, please answer in the chat room for the participants, themselves. And that's the end of the session. And I would like to thank you for your nice presentation, and for the appreciation. Please, the committee share the drawing, and the drawing produced by the local artist from the East Java province for you, Professor Sen.
Can we take a photo with everybody?
Yeah. Okay.
They are doing.
That's for you, Professor Sen. And thank you.
Thank you very much.
Yeah. Thank you very much for your sharing. And now I will give back the time to the committee to continue. Thank you.
Thank you.
Thank you very much, Professor Insinyur Lucas Susanto, MS PhD, for leading the first conference sessions. Looking forward to seeing you again tomorrow, Ivan, the second day of this conference. Well, after this, we will watch together the final poster that has been successfully entered the final stage of Plant Protection Competition 2021 with the theme Innovation and Development Strategy of Phytopathology toward Indonesian Food Security. Now I will guide you all to enjoy it one by one. The first finalist, Rizka Dwi Yulian from Universitas Negeri Surabaya with the title Ruang PA, Platform Pengembang Fitopatologi untuk Penunjang Ketahanan Pangan dan Kesejahteraan Petani di Era Digital. The second finalist, Oki Ahmad Effendi from Politeknik Negeri Malang with the title Irigasi Modern Guna Optimalisasi Pengairan Pertanian dengan Teknologi Presisi di Era 4.0. The third finalist, Shelfa Salsabila Zahira from Universitas Brawijaya with the title Ekoper, ekoenzim sebagai pestisida ramah lingkungan. The fourth finalist, Novandi Rizky Prasetya from Universitas Brawijaya with the title Mycobiosida sebagai pengendalian penyakit antraknosa. The fifth finalist, Ajeng Sayyadatul Maghfirah from Universitas Gadjah Mada with the title Manisan Panambai. The sixth finalist, Dea Putri N from Universitas Gadjah Mada with the title Pemanfaatan Trichoderma spp sebagai agen penginduksi ketahanan tanaman cabai terhadap jamur Coletoricum spp penyebab penyakit antraknosa. The seventh finalist, Wildan Safaat from Universitas Gadjah Mada with the title Tetragen. The finalist, Hoirum Triwahyuli from Universitas Brawijaya with the title Taniku. The main finalist, Verlin Clarencia from Polytechnic Negeri Bali with the title Smart Greenhouse, Work Less and Value More. The 10th finalist, Aprilia Haryanti Gultom from Universitas Brawijaya with the title Kobojan. The 11th finalist, Salsabila Fitri Alfani from Universitas Brawijaya with the title Trichophan. Wow, let's now please give virtual applause for the all finalists. It's a great virtual poster, right? And for you to know more about the plant protection competition 2021, actually the committee already prepared a special documentation that we can enjoy together. And now let's check it out.
Assalamualaikum warahmatullahi wabarakatuh.
Sesi Anda, PT Pupuk Indonesia Persero, PT Medion Farma Jaya, Krisan Catering, PT Multi.
Selamat datang bagi para peserta masih baru dalam
Selamat datang kegiatan ini dan juga saya berharap diskusi ini akan tetap menjadi ikon untuk
itu ada di bawah sehingga kalau bisa seimbang lah gitu. Ini justru mungkin yield loss.
Kajian Korean Biological Agent, pemanfaatan bakteri sebagai pengendali penyakit tawar daun bakteri yang dimana kami dari Universitas Brawijaya.
Gini. Kecil mbak, ininya font di bawah. Begini sih, ada beberapa yang kita butuh malah kecil. Itu judulmu adalah pupuk dan pestisida nabati coba. Tapi kamu sisipin Trichoderma di situ.
Ada 11 tim. Jadi nanti ada 11 kelompok yang akan bermain dalam PPC 100.
Potensi teknologi nanobiofengisida dari seresah daun cengkeh atau Syzygium aromaticum L dalam pengendalian penyakit karat daun kopi, Hemililia fastatrix, V. ETBF untuk mewujudkan pertanyaan. Namun ini masih berdasarkan dari citation dan juga riset yang dilakukan oleh beberapa lahan.
Upaya mengurangi inokulum awal penyakit hawa daun kentang dengan isolasi agensia hayati Trichoderma sp menggunakan media nasibasi.
Media pemanfaatan konus pinus terinduksi Pseudomonas fluorescens dalam media tanam penghambat busuk lunak pada porang sebagai inovasi green technology.
Ecovirus sebagai fitokontrol penyakit plasmatik, Virology orizae, Telesmop magnopod orizae. Dengan subtema pengembangan biotechnology dalam fitopatologi.
Firdaus dari Institut Virologi 10 November akan mempresentasikan karya kami yang berjudul Pemanfaatan nanokitosan dari cangkang udang dan sisik ikan sebagai induksi respon pertahanan Orza sativa terhadap Rhizophonia sativa.
Saya Gabriela S. Mycology dari Universitas Brawijaya. Di sini saya ingin memaparkan mengenai pemanfaatan ekstrak volatile organic compounds atau VOC alang-alang Imperata cylindrica sebagai solusi inovatif pengendalian hama dan penyakit tanaman yang disebabkan oleh kutu daun atau spesies alami.
Pemanfaatan dan peningkatan eksistensi underutilized plant sebagai media alternatif alami perkembangbiakan cendawan Trichoderma sp penunjang ecological agriculture.
Tapi apakah kalian tahu bahwa membuat PTI itu bisa juga dengan cara yang murah?
Jadi apa sudah ada studi gitu bagaimana
kentang yang dipakai itu pun hanya kita bisa pakai dengan cara yang murah karena kentang itu dipakai sebetulnya hanya airnya.
Generasi hebat.
Siap kembangkan fitopatologi
dalam mewujudkan ketahanan pangan Indonesia.
Well, ladies and gentlemen, this competition submission starts from 6th of August until 21st of October 2021, and the presentation itself is held on 23rd of October 2021. The participants come from undergraduate and diploma students all around Indonesia. To know more about this competition and also the poster itself, you can see it through Instagram of PPC Himapta FPUB. And after this, we will have a short break before we continue to the next conversation. For Muslim participants, you can perform Salah Jum'ah or Dhuhr prayer. And we inform you the break will start at 11 until 1 p.m. Western Indonesian time. Hope that everyone will back on time. And while resting, we already prepare several special video compilation. So please enjoy and see you again after the short break.
Kenapa perlu isolasi secara total? Isolasi diri di rumah adalah salah satu langkah awal kita untuk terhindar dari penyebaran virus. Hindari pulang kampung. Tahukah kalian, hindari untuk berpergian jika itu tidak terlalu penting. Pentingnya karantina. Jika kalian mengalami gejala demam, batuk, sesak nafas, hal yang pertama kalian lakukan adalah jangan panik. Hubungi hotline Satgas COVID-19 UB di nomor 0341 403 000 atau 0811 3631 611 atau bisa di nomor 0821 3916 8027. Lakukan karantina atau isolasi diri di rumah atau kos guna penyembuhan. Karena dengan ini kita bisa mendapatkan penanganan yang aman dan tentunya memutus rantai penyebaran virus COVID-19. Selalu jaga kesehatan dan selalu cuci tangan. Iklan layanan masyarakat ini dipersembahkan oleh
Sebagai negara agraris yang memiliki sumber daya alam melimpah, dengan kerja keras bersama memberikan sinyal kuat, sektor pertanian Indonesia semakin kompetitif di kancah internasional. Seminar ilmiah internasional dan Kongres 26 Perhimpunan Fitopatologi Indonesia PFI 2021 dengan tema Peran Fitopatologi dalam mewujudkan tanaman sehat menuju Sustainable Agriculture di era revolusi industri 4.0. Kerjasama Perhimpunan Fitopatologi Indonesia dengan Fakultas Pertanian Universitas Brawijaya, Malang. PT BC Internasional Tbk merupakan salah satu pemimpin dalam industri benih hibrida untuk jagung, padi, dan tanaman hortikultura. Lini produk termasuk salah satu pemimpin pasar untuk produk pestisida dan pupuk non-subsidi. Didirikan pada tahun 1983 oleh Charon Pokpan Group, Go Public, dan tercatat di IDX pada bulan Mei 2007 dengan kode emiten BC. Sebagai perusahaan berbasis sains, PT BC mempunyai fasilitas riset yang tersebar di hampir seluruh wilayah Indonesia. Penerapan teknologi pemuliaan tanaman dengan menggabungkan penelitian lapangan dan laboratorium mampu menghasilkan produk benih yang berkualitas, beradaptasi luas, tahan hama penyakit, dan berproduksi tinggi. Sarana laboratorium bioteknologi PT BC terdiri dari laboratorium proteksi tanaman, kultur jaringan, fisiologi tanaman, molecular breeding, dan genetic engineering. Produk bermutu karya anak bangsa, benih tanaman pangan, benih hortikultura, pestisida, CPT, dan pupuk sebagai produk unggulan petani Indonesia. Pertanian berkelanjutan merupakan suatu platform penting dalam usaha membangun pertanian lebih baik. Praktek pertanian konvensional secara terus-menerus telah menyebabkan polusi air dan tanah, menurunnya keanekaragaman hayati, serta berkontribusi terhadap perubahan iklim. Praktek pertanian berkelanjutan memiliki metode yang dapat memperpanjang nilai guna dari lahan seperti kesuburan, ketersediaan air, serta dapat menurunkan tingkat polusi pertanian.
Konsep berkelanjutan bertumpu pada 3 pilar, yaitu ekonomi, berorientasi pada tingkat efisiensi ekonomi, daya saing, juga besaran dan pertumbuhan nilai, termasuk dalam hal keuntungan yang didapat. Sosial, berorientasi pada kesejahteraan yang dicerminkan pada kehidupan sosial yang harmonis. Ekologi, berorientasi pada terpeliharanya keragaman keanekaragaman hayati dan daya fleksibel atau sumber daya genetik, sumber daya air dan agroklimat, sumber daya tanah, serta kesehatan dan kenyamanan lingkungan. Pembangunan pertanian berkelanjutan bertujuan untuk meningkatkan pendapatan dan kesejahteraan masyarakat tani secara luas. Melalui peningkatan produksi pertanian secara seimbang dengan memperhatikan daya dukung ekosistem, sehingga keberlanjutan produksi dapat terus dipertahankan dalam jangka panjang dengan meminimalkan terjadinya kerusakan lingkungan. Peningkatan produktivitas dan mutu hasil pertanian semakin penting. Hal ini diawali dengan penggunaan benih bermutu. Dalam pertanian modern, benih sangat berperan sebagai paket teknologi unggul bagi petani.
Peningkatan produktivitas dan mutu hasil pertanian sangatlah penting, dan hal tersebut dimulai dengan penggunaan benih yang bermutu. Oleh karena itu, ketersediaan benih bermutu di tingkat petani sangat mutlak diperlukan. Di dalam pertanian modern, di era industri 4.0 saat ini, benih berperan sebagai paket teknologi unggul bagi petani. Benih bermutu juga terus berkembang dan dapat tersedia secara tepat baik dari segi varietas, mutu, jumlah, waktu, lokasi, serta harga bagi petani.
Peningkatan produktivitas dan kualitas hasil pertanian semakin penting. Hal ini diawali dengan penggunaan benih bermutu yang bersertifikat. Ayo, terapkan pertanian berkelanjutan agar swasembada pangan bisa terus dipertahankan. Mau pertanian Indonesia berjaya? Yuk, selalu bangga buatan Indonesia. Persembahan dari PT BC International Tbk.
Hidup itu penuh dengan kode. Ada kode yang bunyinya agak. Ada juga yang bunyinya kode yang cuma satu gaya, tapi punya banyak makna. Kode yang ngomongnya A,
aja, terserah.
tapi artinya B. Kode mata-mata. Kancil 1, kancil 2, atau kode mata. Kode yang bikin seneng. Atau yang bikin ribet.
Nomor rekening lu berapa?
Apapun kodenya, kode buat transfer dibikin simple aja.
Pakai QR code ya?
Oke.
Tinggal scan QR, transferan beres.
Udah transfer ya?
Udah masuk nih.
Dengan QR Code dari BCA Mobile.
QR.
Universitas Brawijaya is one of the most favorite campus destinations in Indonesia. Students come from all over Indonesia and abroad. The campus was established in 1958 by the name of Yayasan Perguruan Tinggi Malang. Located in the downtown of Malang City, which is well known for its fresh and cool air, Universitas Brawijaya creates a beautiful campus environment. Supported by a well-maintained campus area and qualified lecturers, both Indonesians and abroad make the university as a pleasant and conducive campus for teaching and learning process, as well as to develop students' interests and talents. Universitas Brawijaya opens the widest opportunities for prospective well-talented students, both domestically and abroad, including disabled. Currently, thousands of students earning scholarship from various institutions, both the government and private sectors. Universitas Brawijaya has 15 faculties and 2 programs of postgraduate and vocational. Supported by more than 140 professors and more than 2,000 well-experienced lecturers, researchers in teaching and research of various fields of science, technology, social, culture, and humanities, Universitas Brawijaya is ready to develop high-quality and international standardized educational process to produce graduates with high global competitiveness. In order to support students' competence and skills, Universitas Brawijaya provides diverse laboratories with sophisticated and modern equipment, including field laboratories, so the intense research culture continues to grow, indicated by innovative inventions that is useful for wide communities. Excellent scientific works and innovations are created by our academic communities as a means of education, research, and community service programs. As manifestation of its commitment to be a world-class entrepreneurial university, Universitas Brawijaya provides diverse infrastructures and equipment for diverse students' activities. This is an assurance of excellence and well-achieved learner profile produced. Lots of our alumni currently are in influential positions and have real contributions both in Indonesia and worldwide.
Dari pengajarnya dulu zaman saya kuliah banyak praktisinya, jadi saya ngerasa benar-benar kepake banget, bukan cuma belajar teori tapi prakteknya kita dapat.
Dari pengalaman kuliah yang paling berkesan ya karena saya anak organisasi, jadi ya di kepanitiaan, terus saya di sana belajar tentang kepemimpinan.
Dulu yang depan saya itu Fakultas Peternakan. Sekarang udah berapa lantai udah ya? Malah ada liftnya. Dulu nggak ada.
Passionately, we, Universitas Brawijaya, will show the world an education with integrity and harmony at the forefront in science and technology development. Proudly and confidently, the graduates of Universitas Brawijaya are ready to welcome the future, building up the noble future.
Assalamualaikum warahmatullahi wabarakatuh. Now, welcome back again in International Conference and the 26th Congress of Indonesian Phytopathological Society. Still with me, Khairunisa, who will be your companion until the end of this international conference. Hope that everyone here feel recharged after a short break, because after this, we will continue to the international conference with full of energy. And we also have an important information regarding the Plant Protection Competition 2021. So tomorrow there will be an announcement about the winner, but there will be a special category, the most favorite poster. So right now I would like to ask you all to vote your favorite poster through www.menti.com, and you can find the link on your Zoom meeting in the chat column. There will be a committee that send the link there. So hope that everyone here will vote there because it's free, and let's do the vote now. Well, ladies and gentlemen, in the first session, we are already heard the presentation from Professor Roger Koide, PhD, from Brigham Young University, United States. Then from Professor Wee Chang-shen, PhD, from National Taiwan University. And after this, we will hear the presentation from Dr. Insinyur Siwi Indarti, MP, from Universitas Gadjah Mada, Indonesia, and also Professor Nick Talbot, PhD, from the Sainsbury Laboratory, Norwich, United Kingdom. And the second session of the conference will be led by our moderator, and I would like to read her curriculum vitae. In 1999, she has completed her undergraduate studies at Universitas Gadjah Mada, Indonesia, majoring in Phytopathology. In 2002, she has completed her master's studies at Universitas Gadjah Mada, Indonesia, majoring in Phytopathology, and in 2012, she has completed her doctoral studies at Tokyo University, Japan, majoring in Agriculture and Technology. Her research focus are plant host fungal interaction, and then plant-induced resistant agents, fungal pathogen, head shock-induced resistant, and also fungal resistant agents fungicide. And her work experience on 2005 until now, she is a lecturer at Department of Plant Protection in Faculty of Agriculture, Universitas Gadjah Mada, Indonesia. In 2019 until now, she is a chair of PFI Komda Jonglo Semat, and in 2021 until now, she is a chair of Study Program Magister Fitopathology in Faculty of Agriculture, Universitas Gadjah Mada, Indonesia. She also has a lot of track record in making scientific publication and research both on national and international scale, and she also received many awards and grants both on national and international scales. And now, let us welcome Miss Ani Widyastuti, PhD from Department of Plant Protection, Faculty of Agriculture, Universitas Gadjah Mada, Indonesia.
Okay, thank you very much. I hope my voice is clear. Okay, honorable speakers, distinguished ladies and gentlemen, good afternoon from Yogyakarta, Indonesia. I am Ani Widyastuti from Universitas Gadjah Mada, Yogyakarta, and also already mentioned, I'm from IPS, representative Jogja, Solo, Semarang, or we call it as Komda Joglo Semar. I feel honored to serve you as a moderator for the next two presentation sessions. First, we will have a presentation from Dr. C.V. Indarti. She is my colleague from UGM also, entitled Towards Sustainable Agriculture Through Management of Plant Parasitic Nematodes. And here is the short bio of Dr. C.V. Indarti. Dr. C.V. Indarti is lecturer in the Department of Plant Protection, Faculty of Agriculture, Universitas Gadjah Mada. She is plant nematologist and the major of biological control plant nematode interaction. And now she is head of nematology laboratory and also head of agriculture and food division center of agrotechnology innovation, Universitas Gadjah Mada. Dr. C.V. Indarti is also member of Indonesian Phytopathological Society and Indonesian Entomology Society. And okay, I hope Bu C.V. has already been here. So dear participants, we have totally about 45 minutes, and we will have 30 minutes for Dr. C.V.'s presentation, and after that we will continue for the discussions about 15 minutes, so it's quite short. And please drop your questions if you have during the presentation of Dr. C.V. Indarti. Okay, Bu C.V., good afternoon, Bu C.V. Wait, I cannot hear. Sorry.
Okay.
Okay.
You can hear my voice?
Yeah, it's clear now. It's
you.
perfect. Okay. So, Bu C.V., the time is yours for your presentation. 30 minutes, please.
Okay.
Yeah, thank you.
Ladies and gentlemen, good evening all. May I share my presentation first? Okay. As Bu Ani mentioned before, that my my title that I would like to present in the time as Towards Sustainable Agriculture to Management of Plant Parasitic Nematodes. I know all of you know about nematodes, but I think just several people really interested to learn about nematodes in deep. And first, I would like to convey my thank you for the steering committee of international seminar Phytopathology of Indonesia, because you have invited me and gave me opportunity to share about nematodes or plant parasitic nematodes. And the outline that I would present is nematode and plant parasitic nematodes. And the second is nematode problem in Indonesia. And the third, management of plant parasitic nematode toward sustainable agriculture. As you know, all of you, I think, know about the plant parasitic nematode, mostly microscopic, and then sometimes called as a earthworm or roundworm, because the form of the shape is round, like a worm, and sometimes also called as a thread worm or like thread. Nematode has a pseudosolomate, and plant parasitic nematodes or PPN are small, like worm, as I mentioned before, and also multicellular, free-living, or parasitic, which are ectoparasitic, endoparasitic, semi-endoparasite, or sedentary on plant tissue. Usually, PPN or plant parasitic nematode are often not as important as some other biotic constraints, such like pests from insect or other pathogen on crop production in the tropic. But actually, PPN can cause extensive damage and substantial yield losses. PPN are associated with very important agriculture crop and represent as one of significant constraint and global food security. Usually they cause a substantial problem to major crop throughout the world, including staple food, vegetable, fruit, and other plants. And the right side, I saw you all, there are root knot nematodes. The first one in the upper is upper side is a larvae. Second, third larvae, and the lower side is the female root knot nematodes. And root knot nematodes is very destructive, especially on tropical country as such like Indonesia. Crop damage by PPN is not easy to detect because in most cases, it often remains hidden by the many other factors limiting plant growth. As in the slide in the right picture, we saw the coffee plant that attacked by Rhabdobolus similis with symptom damage like unhealthy and yellowing as abiotic factor causes. And farmer and grower, especially in Indonesia, identified insect pests and other constraints of production problem, but usually overlook plant parasitic nematode because the symptoms of nematode damage are not specific resemble with the symptom of other pathogen or abiotic stresses, such as water and mineral deficiency. Plant parasitic nematode not only cause damage individually, but sometimes form disease complexes with other microorganisms and increase the crop losses. Uncertainties concerning the validity of nematode species will lead to practical problems related to the quarantine measure and nematodes management. And the direct or indirect damage caused by various species of plant parasitic nematodes results in delayed maturity, toppling, and also reduced yield and quality of crop produce, high cost of production, and therefore loss of income. In the slide, I took a picture, two kinds of a crop. In the right side, in the left side, this is the pepper that attacked by yellowing diseases that caused by plant parasitic nematodes, especially burrowing nematode, Rhodopulus similis, and combination with the Fusarium. And the right side, this is the onion that attacked by root rot nematodes, Diethylenchus dipsaki, and combining with Fusarium attacked. So that I mentioned before, that sometimes nematode or plant parasitic nematode as an incitant or opening the the infection by another pathogen. And nematode diseases are difficult to control because they hidden nature and hence more often overlooked. Sometimes or some reference said that if plant parasitic nematodes have that is mean entering in the land is cannot controlling because their size is very small and cannot see by my eyes or our eyes. Plant parasitic nematode not only cause damage individual, but usually disease complexes with other microorganisms and increase the crop. And this is the news from Japan in November 2020 that the product from Indonesia, especially from root or material plant that will enter to Japan will reject it because they found Rhodopulus similis in some sample that entered to Japan. So all of material, plant material, will rejected to enter to Japan. And how the impact of plant parasitic nematode on crop? Recently, 4,100 species have been identified, but I think Indonesia not less. Less than 20 species have identified. So collectively, they cause estimate 80 to $1,018 billion per year in damage to crop. Of all the important plant nematodes, the most suspectful species are the sedentary group, which are established and makes a permanent feeding site in the inside of a plant usually in root of plant. They will obtain nutrient while completing their life cycle. In the world, they have top 3 of important plant parasitic nematodes. The first one is root knot nematodes, or myeloidogene SPP. And the second is cyst nematodes, including Heterodera and Hyglobodera. And the third is root lice nematodes, or Platylenchus SPP. Why they as 3 of important plant parasitic nematode? Because most economically and scientifically important due to their relationship with the host plant, also with the white host range, and the level of damage by nematode infection. With the white host range, is cannot manage using crop rotation because they have several or many hosts for infection. How about the nematodes problem in Indonesia? In here, I will share about root knot nematode, Mycology SPP, and also potato cyst nematodes, Globodera rostusensis, the main nematodes, plant parasitic nematode pest in Indonesia, and also endoparasitic nematodes, or Platylenchus and Rhodopulus SPP, and also Hismanula. And then I also share about a white tip nematode, Epelenchoides species, and also root and bud rot nematodes, Diethylenchus SPP. But actually, we have more than 5 genera about plant parasitic nematode in Indonesia. But in here, in the time, I just want to focus 5 genera in Indonesia. Root knot nematodes, by 2006, actually there are 92 species had been described, and half about them was described during the past 2 decades. But in Indonesia, just 6 species have identified, and the problem, or I mean the nematodes, have distributed from Sumatra, and then Java, also Bali, Kalimantan, Sulawesi, and also in Irian Jaya. The species that have found in Indonesia is Mycology incognita, and then Mycology javanica is the first nematode found in Indonesia in maybe in the year of 1040, sometime like that. And Mycology graminicola was found the first time in Indonesia in 1996, I think. And then Mycology hapla, and also Mycology arenaria, and Mycology palax. Root knot nematode also found in seed potato, and because the infected on seed can distributed by seed potato from one area to a new area. And my student investigation on 2018 and published in 2019, we found some species of root knot nematode in central of potato production in Central Java, and also another district. This is the symptom. Makes the seed of potato abnormal. I means the form of seed is with looks like ugly, and in the seed, inside seed, there are some what does mean? Some female of nematodes with the red cycle in here. Some red cycle. We can find some individual of female, and each female can produce it's around 200 eggs, and you can imagine how the nematodes can distribute it from another area to a new area, and as seed as infected, I mean, as a source of infection. And we also found that in every area of center potato production, almost district have a problem with the seed potato infected with root knot nematodes. And here you can saw, let's say in Ngaplak Magelang, 88.23% of sample was infected by root knot nematodes. And then in Kledung, also in Kejajar, and also in Banjarnegara is not. So especially on Magelang, all of seed potato was infected by root knot nematodes. And the second important plant parasitic nematodes is potato cyst nematode, as usually as known as golden cyst nematodes. In the world, we have 6 species of cyst nematode. They are Globodera rostosiensis, Globodera pallida, Globodera tobacum, and Globodera glycine. And in Indonesia, we just have one species. This is Globodera rostosiensis. This nematode was the first time found in East Java, Brasseng, and then distributed to Central Java, West Java, and also in Sumatra, North Sumatra, and recently also found in Sulawesi. And its district, the area distribution of potato cyst nematode also increased. This is the picture of potato that attacked by Globodera rostosiensis. The picture I was took in Central Java, especially in Banjarnegara and Dieng, with unregular symptom and patches of plant growing potato plants. And in the under picture, you can see the female of potato cyst nematode with the red cycle, and they are in, I mean, in about 5 centimeters of fruit. They can, we can find, I can found some individual of female of potato cyst nematode. And this is our publication when we found the first time of potato cyst nematode in Indonesia in East Java. And also we can saw in here that several damage caused by potato cyst nematode, Globodera rososiensis, can show yellowing, and I think will cause reducing growth of plant. And we also investigation in a growth chamber in my faculty or my department, and we found this is the yellowing color of a cyst, and its root can we see in here. And the third nematode, important plant parasitic nematode in Indonesia is migratory plant parasitic, endoparasitic migratory. There are some species, Pratylencus. We have Pratylencus coffee, usually attack on coffee and cause almost dead seedling in East Java also. And also Rhodopulus. Usually we found, or mostly found, Rhodopulus in coffee also, and also in banana. And coffee, usually we found in coffee Arabica. They are grown in in highland area. And the other endoparasitic migratory is Hismanila. We have two species. Up to now, we have 2 species of Hismanila in rice, or that attack on rice. The first one is Hismanila oryzae, and the second one is Hismanila murconata. This is also a new species that found in Central Java and Yogyakarta that attack on rice also. And the other relative new species of plant parasitic, they are Aphelencoides species or white-tape nematodes that attack on rice and also include as economic important for rice plantation. And here we found from 70 samples, we found 16 that positive attack by Aphelencoides species. And here you can see that all of seed rice or rice seed have been certificate from company, but they also have a problem. So as I mentioned before, that usually plant parasitic nematode is neglected or not have attention more. And in here, in its 10 seed of rice, we can found some population of Aphelencoides species in its seed of rice. And here you can saw like this. And rice itself, we have not only root knot nematode, not only his manila or white tape nematode or Aphelencoides species, but rice also attacked by Trichomonas and also Helicotylencus and Cratylencus also. So for plant parasitic nematode that have a wide range of host plant, I think it's difficult to control. The next plant parasitic nematodes that I explained about the Diethylencus dipsaki. This is the stem and pulp rot nematodes, usually found in garlic. And for the first time, we found in Central Java and Temanggung. And we found some, some varieties of garlic also as a host plant for Diethylencus dipsaki. The symptoms with the necrotic and then rot on bulb and also on root and cause plant growth. What does it mean? Plant growth like this. Okay, I will continue. This is the symptom of Diethylencus dipsaki attack on garlic with the distortion of leaf and rot with the puff. Usually we found also association between this nematode with the fusarium. And this is also another area that have a problem with Diethylencus dipsaki and on garlic also. The picture I was took in another district in Central Java. And the third section, I would like to share about the plant parasitic nematode management toward sustainable agriculture. There are three components. The first one is prevention by exclusion or avoidance, and also by limiting the spread, especially of quarantine of plant parasitic nematodes. There are 63 of A1 quarantine plant parasitic nematodes, and for A2 we have around 10 species as a quarantine plant parasitic nematodes. And PPN control, we can apply some technique such like implementation of control by cultural technique, by physics or mechanic, and also by rotation of plant, especially host plant, and also habitat management or habitat engineering, and then by biofumigant. Because biofumigant is now a trend for controlling plant parasitic nematodes, especially for sustainable agriculture that are environmentally friendly. And
so we apologize.
Yeah.
Could you make shorter, please? About 2 minutes.
Okay. Thank you. Yeah.
Okay,
Thank you.
Bu Siwi.
Thank you for your reminder, Bu. And the Okay, because I have some slide. I still have some slide. And the third component is reduces nematode status safe avoidance. And this is I will save to another slide. Prevention by certification of seed and propagation. I think there is no available in Indonesia. I don't know the other country maybe have one. And method implementation by sanitation, cultural technique, host resistant and biological control and chemical. In here, recently, I with Pak Setio Hartono start to implementation of solarization to reduce plant parasitic nematode and other pathogen. And here we saw that by solarization, the temperature of soil can increase up to 47 degrees. Biological control, we also have experiment by Nematophagous fungi and also by Bacillus. For Nematophagous fungi, we have also some experiment, such like Nematophagous fungi for controlling potato cyst nematode. You can you can see in here by application of Nematophagous fungi can reduce the population of cysts of potato cyst nematode, and also application of Nematophagous fungi can also improve the performance of a plant, such like plant height and also yield. And on the other hand, in here, Bacillus is also can use as a biological control for controlling potato cyst nematodes by what is mean enzyme that are cause death of cyst and also of eggs of potato cyst nematodes. And my conclusion, plant parasitic nematode management towards sustainable agriculture should be supported by education for, I'm sorry, maybe this is a education for farmers, especially about plant parasitic nematode, and also study of economic threshold to economic important devastating species of plant parasitic nematode, and also strengthening research collaboration and combining multidisciplinary experts working on nematology is required to combat the economically devastating of nematode in Indonesia, especially for area mapping of distribution and also identification of indigenous species, because by change of field using can increase the safe of indigenous species become important, economic important to plant parasitic nematodes. I think it's enough, Bu Ani. Thank you very much for ladies and gentlemen for your attention, and thank you very much again. And I to Bu Ani, come back to Bu Ani. Thank you.
Okay, thank you very much, Bu Siwi. It's really wonderful and important presentation. Even we got many new information, Bu Siwi. Even for me personally, I just realized that in rice field, not only one species, not only two species of nematode can attack the rice plant. Yeah. And we have already got some questions, Bu Siwi, because this topic is really, really important. First from Dr. Shibli. Yeah. Yeah. the question is, I have a question about interaction endophytic bacteria, endophytic bacteria, and nematode. Is there any reports about that? So the interaction between endophytic bacteria and nematode, as nematode will suck the nutrient flow from the root tissue. The hypothesis is the endophytic bacteria might be also flowing to the guts of nematodes. So the bacteria also flowing to the gut of nematodes. Is that possible that endophytic bacteria are able to control parasitic nematodes? So about the biocontrol of parasitic nematode using endophytic bacteria, Pusiwi. Yeah, please, Pusiwi.
Okay, thank you. I think it's a great question from Dr. Sibli. Yeah. There are many information, paper that have published, especially from IPP, that have investigated about endoparasitic, endophytic bacteria with plant parasitic nematodes. Yeah, maybe Pak Sibli can searching about the paper about the endophytic bacteria can controlling plant parasitic nematodes, especially on root knot nematodes and other endoparasite nematodes. Okay. I hope it's clear.
Yeah. So, okay, thank you, Bu Siwi. And we will move to the next question from Bu Heni, Sri Widinugraheni to Bu Siwi. To your experience, what was the best method to control the nematodes on banana plantation, Bu Siwi?
Okay.
Yeah.
Okay. On banana plantation, I don't have experience in controlling plant parasitic nematodes on banana, but I have experience about experiment about identify what's the nematodes usually attack on banana, and I think for banana we can use free seedling. Nematode free seedling by hot water treatment combined with nematicide for treatment corm of banana from infected area if farmer want to plant to another area. Also, we can use maybe antagonistic plants such like tagetes can use for controlling and controlling plant parasitic nematodes by plant in the row between banana row and banana row 1 and row and another row. We can plant what does it mean? Tagetes. Okay.
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 Still any information specific about the banana or enough?
I think it's enough. Yeah.
Okay. Yeah. Thank you very much. Yeah. Nice information, Pusiwi. Tagetes is so tagetes is powerful against nematodes. Yeah. Okay. Nice. Thank you. So we will move to the next question from Bapak Muhammad Iqbal Maulana. Yeah, I have some question for plant parasitic nematode management in Indonesia. For example, Meloidogyne or Globodera have crops cultivar carrying resistant genes applied as management strategy. The first question, and if so, do we have local Indonesian cultivars in which the R genes comes from? So it's about the resistant cultivars, resistant genes, Pusiwi. Do you have any information about that?
Yeah, I think in Indonesia, as I mentioned before, there is no nematologist work in what does mean about the plant resistance, but in my PPT, I presented about that just start. We're looking for the gene resistant from what does mean? Solanum from tomato and also eggplant. We just start to identify what the gene have resistant to root knot nematode. But for potato, I think there is no information in Indonesia, but some cultivar, such like Atlantic, is I think a moderate resistant to potato cyst nematode.
Okay. So I think it's very interesting topic to see about the resistant genes, as we know that in Indonesia we have so many like a local variety of the vegetable. So I think it will, maybe just we need more nematologists, right?
Yeah.
To work together.
Yeah, Bunny, I think
because
I agree with you.
maybe still many things to do, and also we need the young nematologists. I hope that some students attend in this seminar will also interested at becoming a next generation of nematologists.
Hopefully.
Okay. Okay, Bu Siwi, we continue to the question from Ibu Endah Yulia. I have question about the pest control or management for a case in one slide that's showing some potato plants attacked by nematodes. So from your slide, Bu Siwi, do we need to eradicate all the plants since the plant seems still young? Yeah, maybe from your slides, the plant is still young and already attacked by the nematode. So what should we do?
Usually, the farmer is also still allowing potato growing, but with the yield is lower. And I have an experiment in Dieng, Central Java. They are a farmer group that adopted our technique, but not all farmer, I think, because the implementation about the fungal that I found, and also combining with the what does it mean? Rotation of plant with without potato during 3-time planting. So I think it's not good choice for a farmer.
Okay. So must change the plants. Okay.
Yeah, just the plant at least 3 times without potato plant. So they plants with the flower that have high economy value. That's it. But not all a farmer can follow the methods like that.
Yeah. And also Bu Siwi mentioned the biological control using the fungi. So before, okay, so that's the answer for Ibu Endah Yulia, using biological control, using like a fungal that can also become the antagonist of nematodes, and also the the mixed cropping, mixed cropping, and also the rotation. The
rotation with other plant, not host plant. Yeah,
not host plants. Okay, thank you, Bu Siwi, and we will move to the next question. So, Bu Siwi, everyone in this room is really appreciate your presentation. Yeah, because we think that the information you give us is very important, and we really thank about that. And we still have some questions to CV. Next is from Bapak or Ibu. I'm sorry if I made mistake. Ayi Marsudi. The question is, I have some question. How much female meloidogen is used to be identified on potato bulbs using the pattern perennial method? How much female, so for the identification.
Okay.
Yeah. And how to multiply NSK in the laboratory and the growth chamber.
Okay. There are two questions. The first one is how many females that usually used for identification by perennial pattern. For as I think it's depend on the what is mean. The scale of human resource. In what is mean? In scientific rule, at least 20 females should be measured to morphometric, but for perennial pattern essay, I think just need 3 or 5 female to know the perennial patterns that to identify species level, and how to multiply the answer of 4 to the second question is how to multiply assay. Usually I use egg mass, 1 egg mass, and then inoculate in one plant of such like tomato or chili that planted on sterile media. Yeah. Why 1 egg mass? Because we need the nematode inoculum should be homogeneous. So for one species, usually come from 1 egg mass. Is that?
Okay.
And we need it's around 2 months for producing inoculum of root knot nematodes.
Okay, thank you very much, Bu Sibi, for your answer. And maybe once more question due to the time, or maybe later, Bu Sibi can also check the question in the chat box, because maybe not so many more. So the question that we cannot cover in this conversation, discussion, maybe later we can also answer. But we still have time, so would you please answer one more question from Pak Faiz? Yeah. What's the difference between plant pathogenic nematodes, or maybe plant parasitic nematode, because it's written PPN, and entomopathogenic nematode based on their morphology? Is there any difference?
Okay. According to me, plant pathogenic nematode and plant parasite nematodes is can differentiate or can can divide it by the symptom of host plant. Usually, plant pathogenic will cause what does mean physiologist disturbing, and parasite is just nematode just puncture the cell of plant, and then sucking the content of cell. And however, for plant pathogenic nematodes, usually cause abnormalities and on feeding site or site where nematodes feeding will abnormal of roots such like from giant cell or syncytia or gall like that. And for entomopathogenic nematodes, we can see by the stoma of nematodes. Parasitic nematodes or plant parasitic nematodes have a stylet, but entomogenic nematode they don't have a stylet. It's different because they can cause death of on insect because they release bacteria symbiont that mean insecticidal or insecticidal and can death of insect. This one.
Okay, thank you very much, Bu Siwi, and I apologize to cut the session because of the time. Yeah, actually we have a very wonderful and also important presentation from Bu Siwi, so we would like to express our gratitude, and I think the committee has prepared something special for you. Maybe the committee, please would you please help me to share the painting special for Bu Siwi. Hello.
Not ready, maybe.
Not ready yet. Okay, Bu Siwi, for the certificates, the committee will send the certificate to you later on. But actually, the committee said to me that there is a special painting, and which is made by the local artists from the East Java, who also become the participants in this meeting and also make a painting of you during your presentation. It's wonderful actually, but I don't know why. Really. Yeah, I'm also excited to know about the picture, but I don't know why. I don't know.
It's
so
very excited.
Dr. Shibli said that. Okay, later on we will see. Okay, sorry for that. And once more, we would like to thanks to Bu Siwi for the great presentation, really, and also for all the audience for the active discussion, because we have so many questions during the presentation, and hope we will see you again, Bu Siwi, and we will continue to the next session. So thank you very much, and let's give the virtual applause to Bu Siwi. Thank you, Bu Siwi.
You are welcome, Bu Ani, and ladies and gentlemen. Thank you very much.
Thank you. Okay, so dear honorable speakers and ladies and gentlemen, so I will give the next session first to the Masters of Ceremony, maybe. Is there any information or? Okay. Okay, or can directly we move to the, okay, so we will directly move to the next keynote speaker. Okay, let me first pin because I cannot see the small. Okay, well. Okay, all right. We will continue to the next speaker. Here we have Professor Nicholas Talbot. Professor Nick Talbot is a group leader for the Sainsbury Laboratory, Executive Director. He is a molecular geneticist, and for his career, he accomplished his postdoctoral research fellow in Purdue University, USA, and then he is lecturer, professor in the School of Bioscience, University of Exeter, and head of school, and also the same School of Bioscience, University of Exeter, UK, Deputy Vice Chancellor of University of Exeter, and Executive Director in the Sainsbury Laboratory, Norwich Research Park, UK. Welcome, Prof. Nick Talbot. Good morning, I suppose you there in UK. Is it right? Is it morning?
Yes, it is. It is very early in the morning. 8
early
a.m.
in the morning. Okay, so thank you for attending this seminar, Prof. Talbot, and we are lucky to have you here, and we will learn from you about your presentation entitled Investigating the Cell Biology of Rice Infection by the blast fungus. So I think this is very special day that we learn about blast fungus both in the morning and the afternoon session from you next. And okay, we have the Prof. Talbot short bio already. So without further ado, I give the time and also the screens to you, Prof. Talbot. Yeah, your time is 30 minutes.
Can you see my main slide, or can you see the speaker notes? I don't know which one you have.
Yes, perfect, perfect.
So you have the main screen. That's
Yes, investigating. Yes, the main screen.
Okay. Thank you for allowing me to talk this morning. I'm really very, very pleased to be here. I'm going to tell you about the work that we do on investigating the cell biology of plant infection by the rice blast fungus, Magnaportha oryzae. And I'm going to try and really address these two questions. So how does the blast fungus infect intact host plants? And how does the fungus then invade plant tissue? So how is that? How do those two processes occur? So those are the things I'm going to be talking about this morning. Well, rice blast, of course, many of you will know about rice blast. It's a really important disease across all rice growing regions of the world. It can take up to 30% of the annual rice harvest across the world. So in many years, many people are used to seeing between 10 and 20% yield losses. And of course, it can affect seedlings where you get these necrotic lesions on the surface of rice plants, and in heavy infections, it will even kill rice seedlings. But these seedlings can expand, these lesions can expand over time, and on larger leaves, you can see them spreading to up to 1 centimeter in length. And they'll produce between 20 and 50,000 spores every 24 hours. The fungus will sporulate normally at the dew point early in the morning. And that enables really big inoculum buildup. So rice blast infections can rapidly build into epidemics as a result of the massive release of spores from lesions over several weeks. And of course, the disease that farmers really fear the most is this disease, neck blast, which is when the fungus is spread into the stems and nodes, where it can cause node blast and neck blast and panicle blast. And in heavy infections, you can see this ring of sporulation around the node, and that will rot away the inflorescence, the panicle that carries rice grain, and you'll get very, very severe yield losses. So I took this picture in Hunan province in China several years ago now, and the farmer told me he had about 80% yield loss that year. So this can be really very devastating. So rice blast can affect all rice growing regions of the world, and there's an outbreak right now happening in West Africa, for example, and this is just an example of one which we're familiar with. This is a field trial site where we're carrying out field trials of potentially disease-resistant lines of rice blast that we've bred, and in Burkina Faso in West Africa, and there's a really severe rice blast epidemic going on now. But Magnaportha oryzae, the fungus that causes rice blast, doesn't just infect rice. It actually infects a whole host of different grasses. So as well as Oryza sativa, it will affect many other millets, so foxtail millets, finger millet, pearl millet. So it'll affect many other millet species and many other grasses. So about 50 different species of grass can be infected by Magnaportha oryzae. But the fungal strains that infect those species of grass are normally limited to that host. So there are host specificity determinants both in the fungus and also in the hosts that prevent infection of different grasses. But sometimes the fungus can undergo a host jump, and it's done that quite recently, where it's jumped from forage grasses like Lolium and Echinocloa, and it's jumped onto wheat. And the result of that has been this new disease called wheat blast. Now, wheat blast was first observed in South America, in Brazil in 1985. But a single pandemic clone has actually now spread across the world and spread into Bangladesh in 2016, when it took around 12% of the wheat harvest. And it's got progressively worse in the last 4 years. And I visited Bangladesh in February 2019, and there was absolute devastation. The disease there actually resembles Fusarium head blight and other heading diseases. It's a spike disease. It actually causes a ring of sporulation around the neck, and this will again destroy all of the wheat grain before harvest. So it's a really serious disease, and rather scarily, this disease is now spread into Africa, and it's been observed in Zambia at 3 different sites in Zambia, and we've sequenced a number of genomes from Zambia of the wheat blast fungus, and we can show that this is related to the same strains that left South America. So this disease is spreading across the world very rapidly, and with climate change, it really does threaten wheat production. So Magnaportha is a really serious disease of rice, which many of you will be familiar with, but also now a serious disease of wheat. So 2 of the most important food sources on the planet are both affected by this fungus. So it's a really serious disease problem. The thing that's always interested me about rice blast, though, is this life cycle, the developmental cycle, where 3-celled spores land on the surface of a rice leaf and send out a germ tube. They make a special cell called an aprosorium, which is used to penetrate the rice leaf surface, and then the fungus can spread very rapidly in plant tissue. And that's what I'm going to tell you about today. I'm going to talk through this life cycle and tell you about the biology at different stages of development that we've been studying. So how then does the blast fungus infect host plants? So how does it do that? How does it gain entry to rice plants? Well, what happens is that this 3-celled structure, the conidium, will germinate, and it will send out a germ tube, which then forms this polarized, dome-shaped cell, the aprosorium. And I hope you can see that the edge of that cell is becoming darker and darker. So it's accumulating melanin in the cell wall. And that's really important in conditioning the ability of that cell to generate pressure, so really high pressure. And the other thing I hope you can see is that the spore collapsed. So during that process, all the contents of these 3 cells in the spore ended up inside this one cell. So 3 cells, all their contents went into this single aprosorium. And what you'll end up with is this. So on the rice leaf surface, here's the aprosorium on the leaf surface, and then the spore is now an empty bag. So all of its contents have ended up in this cell. And that's important because that conditions the ability of the fungus to generate really enormous pressure. So this dome-shaped cell will draw water in by osmosis because it accumulates glycerol. So it makes glycerol to very high concentrations, up to 3 molar glycerol inside the aprosorium. That draws water in by osmosis. So water enters the cell. But the melanin in the cell wall prevents that water from leaking out. And as a consequence, the fungus will begin to generate pressure. And the pressure really is enormous. The pressure will generate up to 8 megapascals of pressure. So that's 80 atmospheres, so 40 times a car tire, very, very high pressure. And that pressure is then applied at the leaf surface. So a narrow penetration hypha will rupture the cuticle and it will enter the cells below. And Magnaportha is capable of breaking thin plastic membranes. So it can use physical force to break the rice cuticle. So it also secretes enzymes at that interface, but we know that even inert surfaces can be broken by the fungus just using sheer force. Now, what are the prerequisites for making an aprosorium? Well, there are 3 things that need to happen. The first is this. This is a pathway which monitors the leaf surface. So these aprosoria will only form on a hard hydrophobic surface. So the fungus is able to tell whether the surface has those characteristics using a series of surface sensors. And those coalesce around this signaling pathway, which is called the PMK1 mitogen activated protein kinase pathway. And what happens is that there's a succession of phosphorylation events leading to phosphorylation of this protein called PMK1. And PMK1 is a MAP kinase, which then regulates a whole series of transcriptional regulators. And they regulate the formation of this special cell, the aprosorium. So if you take away this protein, if this protein is deleted, the gene encoding is deleted, you end up with this non-differentiated germ tube. So it can't make this cell. So unless it has this PMK1 gene, this cell doesn't form at all. You just get an undifferentiated germ tube. And what's actually happening inside the inside Magnaportha is a huge change in gene expression. So more than 4,000 genes ultimately are differentially expressed as a result of going into aprosorial development. So we know that if we look at the fungus forming on a hydrophobic surface, there are around 4,000 genes which are responsive to that hydrophobic surface. And of those, 3,500 of them are dependent on PMK1. And we now know that there is a series of transcriptional hierarchy of regulators which enable this massive change in gene expression. So the fungus is actually turning over around 40% of its genome as a result of making this aprosorium. So making an aprosorium is a very complex process involving around 4,000 genes. So it's a complicated process, but it's regulated primarily by this very important PMK1 pathway. And we've just published a paper. It came out yesterday. So if you want to look at that, it describes the mechanism in which that happens. The other prerequisite is cell cycle control. So the conidium has got 3 nuclei. There are 3 cells, and you can see the 3 red nuclei. And one of them divides here, a mitosis happened, and entered the forming aprosorium. Then all 3 of those nuclei in the conidia will degrade and die. So you can see that when Magnaportha infects a rice leaf, there are always 3 nuclei in a spore. And the 3 nuclei become 4 nuclei and then become 1 nucleus. And that 3-4-1 pattern always happens. Every single infection, every time the fungus infects, it's completely invariant. And that's because the process of making an aprosorium depends on the cell cycle. So only if the nucleus that was in this cell enters DNA replication will the aprosorium begin to form. And only when it has entered mitosis will this cell mature and make melanin in the cell wall. And only when it leaves mitosis will the maturation actually be completed. So there's a whole series of cell cycle checkpoints that regulate aprosorial development. And the final prerequisite is that this conidium actually has to die. It must die in order for the fungus to infect rice. So we know that in a wild type cell, we have this single nucleus, so it's gone through 3-4-1 pattern. But if we delete any of the genes associated with this process, autophagy, which is necessary for this cell to be consumed and its contents trafficked into the aprosorium, if any of around 20 genes involved in that process are deleted, then the nuclei can't degrade, they become distended, but they're unable to degrade. But importantly, this aprosorium can't cause disease, so there's no rice blast in the absence of that. So essentially, those are the 3 important prerequisites. They have to occur. There has to be the PMK1 pathway to regulate perception of the hydrophobic surface. It has to undergo mitosis through the cell cycle, and the conidium has to die. And if those 3 processes happen, then rice blast will occur. So what happens next? So here's the aprosorium on the leaf surface, and here's the base of the aprosorium where you can see there's no melanin. The melanin layer is in this dome of the aprosorium, but here there's a very, very thin layer. And what happens is at the base of the aprosorium, right in the middle here, the fungus will undergo this repolarization. So it will form this peg, and this is rupturing the cuticle of the rice leaf. This is the penetration peg or penetration hypha. And what we now know is what's happening at the base of the aprosorium is formation of this ring structure, which is made of actin. So filamentous actin is forming a toroidal structure, a donut-shaped structure, a ring at the base of the aprosorium. And that's enabling the fungus then to repolarize, to enable it to form that peg. So the actin has to be remodeled and reorganized into that ring structure. And that requires a group of proteins which are called septins. Now, septins are part of the cytoskeleton. They're cytoskeletal proteins. And during aprosorial development, as you can see in the movie, they form this beautiful ring structure. So that beautiful green ring is a heterooligomeric assembly of 4 different septins. So the septin ring is formed by these 4 septins, sept 3, 4, 5, and 6. And they collectively form this ring structure. And that ring structure acts as a scaffold for actin to form, but also it enables proteins involved in repolarization to be localized in the middle here, at the point at which the peg will emerge. So the formation of this ring structure in the aprosorium is absolutely critical to making the aprosorium functional and enabling it to penetrate a rice leaf. And it also requires regulated synthesis of reactive oxygen species, which are involved in regulating septin assembly. So if you take away the septins, then that beautiful ring structure of actin doesn't form properly. So septins are able to organize that actin network. So if you delete sept 3, 4, 5, or 6, then you just see this tangled mess of actin, and the beautiful ring structure doesn't form anymore. And as a consequence of that, the fungus can't cause disease anymore. You just get a resistant response. And the aprosoria on the leaf surface, which would normally make a penetration hypha to rupture the cuticle, they can't do that anymore. They're unable to do that. So that prevents aprosorial formation occurring. So we know that septins are really important in the context of plant disease. We also know that the septins are able to organize a whole variety of other different proteins. So here are 2 septins, sept 5 and sept 6, forming that ring structure. So when they do so, the actin forms, again, a ring here. And this is an actin binding protein called gelselin, which you can see binds to the inside of that ring. This is a regulator called CHM1, which is a kinase, which is involved in regulating repolarization and septin function. That also organizes to a ring structure. This protein, RVS167, is right in the center of the ring, and it's kept in place by the septin, and that's involved in membrane curvature generation and endocytosis in the center of the aprosorium. T1, you can see again forming a ring conformation, and that is associated with actin polymerization. And XO70 is involved in secretion, so secretion of proteins into plant cells. So it's part of the exocyst complex. And all of those structures are septin dependent in the way in which they accumulate, all at the base of the aprosorium here. And what we found is that that process depends on this cell making turgor. So if the aprosorium is able to generate pressure normally, then this happens. All of these ring structures form. But if we identify mutants which can't regulate turgor properly, then we see mislocalization. And this is a mutant called SLN1. And SLN1 is a mutant affecting a histidine aspartate kinase, which is necessary to monitor pressure inside the cell. And in the absence of SLN1, the fungus can't determine how much pressure it's forming. So in fact, it makes too much pressure, but it never repolarizes. And then when that happens, we can see the septin rings don't form. We just see this, again, tangled mess of septins, but they don't form a ring. The actin doesn't form into a ring structure. Neither does gelselin or CHM1. RVS167 is mislocalized, so is T1, and so is XO70. So none of these processes can occur if the fungus can't monitor turgor correctly. And to show you what happens in a SLN1 mutant, I'm going to play the same movie that you saw previously when you saw that beautiful septin ring occurring. And what you can see is in this SLN1 mutant, the septin ring tries to form, but instead you can see it's unable to form that ring structure, and it forms this patch of septins, which then deteriorate over time and get smaller and smaller. And I hope you can also see that this aprosorium is getting darker and darker and darker. And the reason for that is it's over accumulating melanin. So it makes too much melanin. So it just forms more and more glycerol and more melanin until eventually it will burst, but it's unable ever to penetrate rice leaves. So it can make pressure, but it can't convert pressure into force. And this is a turgor sensing kinase called SLN1. So if you delete that gene, if you can't have SLN1, then the aprosorium becomes hypermelanized and it's unable to cause disease. It can't repolarize. And this leads us to a model which looks like this. So essentially at the base of the aprosorium, we can see this septin ring, and the septin ring forms at the base. That was the green ring that you saw. It organizes actin at the base of the cytoskeleton, and then proteins involved in actin polymerization, which will lead ultimately to this PEG forming. And proteins like LAS17 involved in actin polymerization, and also proteins involved in membrane interactions with phosphatidyl inositol phosphates, which are accumulated at this point. And this leads to the PEG forming and the aprosorium rupturing the cuticle. Now we know what's happening in the cell, and I'm not going to show you all the data because there's too much to show really. But but SLN1 is able to bind to 3 different stretch-activated gated ion channel proteins. They're able to sense the pressure being generated inside the cell, and then under those circumstances, SLN1 will down-regulate melanin biosynthesis, and working with the cell integrity pathway, it will phosphorylate this NOx complex, which leads to the septin ring forming, and also phosphorylate this phosphodiesterase, which will down-regulate the production of glycerol. And it will then lead to rapid glucan and chitin synthesis at the base of the aprosorium. It will lead to septin ring formation, and also the endocytosis and secretory processes required to make this penetration PEG. And all of those are ultimately dependent on septins, which are aggregate in response to turgor. But also, the other thing that we can't forget is that it is also linked to the nuclear state of the aprosorium. So only if the aprosorium is passed into S phase will this whole process carry on and be operative. So that's what we know about aprosoria. So we know that they generate pressure, and we know that that process of repolarizing involves cytoskeletal reorientation, remodeling, and that's dependent on a turgor sensing protein kinase called SLN1. So what happens next? So what happens now when the fungus invades plant tissue? So we've spent all our time so far on the surface of a leaf, so on the outside of a plant. So what happens next when the fungus enters a plant? Well, if we look at that microscopically, there's the aprosorium on the leaf surface. And you can see if you pull away that material, what you can see is these invasive hyphae. And the invasive hyphae have now broken into the plant cell, and they've started to form. But interestingly, they don't look like normal hyphae. They actually resemble budding yeast. So they are able to undergo this budding morphology, where they undergo an axial budding, which leads to them being able to penetrate and grow rapidly in this initial epidermal cell. But interestingly, what happens is, although the cuticle and the cell wall is ruptured, the plant membrane, the plant plasma membrane, is still intact. So this is a rice transgenic line, which is expressing a GFP, which is localized to the plasma membrane. And the fungus is just expressing a cytoplasmic RFP. So this is the fungus growing inside a rice cell, but you can see that the plasma membrane is intact and it's around the fungus. It invaginates around the fungus. So Magnaportha is able to grow inside living rice cells, which have an intact plasma membrane. And interestingly, when it moves from one cell to the next, it maintains that membrane integrity. So this is the fungus. Here's the aprosorium out of focus on the leaf surface. These are invasive hyphae in this cell, and now they've moved into this cell next to it. So you can see they've moved into this cell on the left. And that cell here, which is bound by this, we call this the extra invasive hyphal membrane. It's still intact. So every time it's moved into a new rice cell, the membrane's intact. But as it leaves the cell behind it will lose its integrity and eventually will die. So this is how you get the formation of disease lesions. So the fungus will always move into living tissue, but behind it, the cells will die, and ultimately the fungus will sporulate from those dead cells. So this is a sort of classic hemibiotrophic fungus. It's biotrophic as it's growing, so it always occupies living cells. So it's always biotrophic at its growing margins, but the cells that it's vacating will die, and that will fuel sporulation. So they will end in a sort of necrotrophic manner. So it's a classic hemibiotroph. And that's the way that it is able to move from cell to cell. But we became really interested in this. How does this happen? How is the fungus able to do this? How is it able to maintain the integrity of this membrane as it moves from one cell to the next? And this was the thing that really fascinated us. Well, we know that one way it's able to achieve this is the fungus is able to secrete a battery of effectors. And in this video, what you're seeing is Magnaportha infecting a rice leaf, and this is a whole plant on a microscope stage. It's not a detached leaf, it's a living plant, and this is a living fungus inside the plant. And there are two different types of effectors being made. The green effector, the one that's here that's being secreted around the hyphae, that's an apoplastic effector, and that's destined to be delivered into the space between the fungal cell wall and the plant plasma membrane, which is the apoplast. And then those types of effectors will suppress extracellular immunity, things like chitin-triggered immunity on the outside of the cell. Meanwhile, another group of effectors are exemplified by this red dot here, which is called the biotrophic interfacial complex. And that enables the fungus to secrete effectors directly into plant cells. And they'll end up in the cytoplasm. Sometimes they go to organelles or even the nucleus, but they will be delivered inside of rice cells where they suppress immunity responses. They suppress PAMP-triggered immunity processes, which would lead otherwise to a defense response. And the fungus secretes a whole battery of these effectors. Now, we know by doing genomic analysis that what's happened is that big group of effectors, about 400 effectors, will be secreted. And what that means is it enables the fungus to then grow really rapidly in tissue. So it will grow incredibly rapidly in plant tissue. And you can see that it's now occupied all of those rice cells. This is a three-dimensional reconstruction inside a living plant cell. And the fungus is forming these bulbous branched hyphae inside cells, and then it will move to vascular tissue where it forms these longer runner hyphae. And it will then rapidly fill plant tissue, very, very rapidly. This is only around 50 hours after infection. So you see the fungus moves very, very rapidly once it's inside rice leaves. And again, we became interested in working out, well, how is the fungus moving from one rice cell to the next? How's it able to do this? So we looked at this in more detail, and what we found is that that if we looked at the junctions between rice cells, we saw this process happening. So here there's the fungus, and it's encountering a boundary between two rice cells. And when it encounters a boundary, you can see that there's severe hyphal constriction, and then it moves through that site into the next cell. So it makes a swollen structure, it then constricts and then moves through into the next rice cell. So this resembles that aprosorium development on the leaf surface. And this, as you'll see later on, there are lots of parallels between these processes. The marker that you're looking at here is gelsilin GFP. So this is an actin binding protein. So what you're seeing there is an actomyosin contractile ring forming at the base of the aprosorium, at the base of this infection structure inside the cell. which we now call the transpressorium, as I'll tell you in a moment. It's moving through these sites. So what are these sites? Well, those sites are in fact pit field sites where plasma desmata accumulate, and the fungus is able to detect where they are and move through those sites into adjacent cells. And that's seems to be how it's able to maintain this membrane integrity. So if you look at that process again in a bigger field of view, you can see this is what it looks like in a whole field of view. You see the fungus every time it encounters a boundary, it undergoes that constriction and then moves through into these adjacent cells. So every time there's a boundary, you see these severe hyphal constriction points and it moves through into the next cell. So how is it doing that? How is that process occurring? So to investigate that, we then went one stage further and we decided we're going to do electron microscopy of this region to try and see in more detail what's actually happening at that point. So if you do electron microscopy, and we took multiple sections, we took up to 100 sections through a field of view, and then we built a tomographic reconstruction, a computer reconstruction, you can see the extent of hyphal constriction. So it constricts to a diameter of about 360 nanometers. And that is the same diameter as a pit field. So it's able to push through an entire pit field site and then enable the fungus to move from one cell, this swollen hypha, into the next cell, this other swollen hypha here. And you can see the plasma membrane invaginating around the fungal hyphae. And here's the fungal cell wall that it's ruptured to move, the plant cell wall that's been ruptured in order to move from one rice cell to the next. So how are we going to study this process? So we wanted to work out, well, how is this process regulated? Well, to do that, we went back to our favorite pathway, this pathway. So I showed you this right at the beginning of the talk. So remember, this is the PMK1 MAP kinase pathway, which is required to enable formation of an aprosorium. But what I remembered is that if you delete PMK1, it can't make an aprosorium, so it can't invade, it can't infect plants. But if you wound plants, if you actually remove the cuticle, you abrade the cuticle, or you inject fungal hyphae or fungal spores directly into plant tissue, then I remember that Magnaportha still can't cause rice blast when it lacks PMK1. So a PMK1 mutant can't cause blast disease at all, even when you inoculate wounded plants. So that suggested to us that this protein kinase must also be involved in events after penetration, not just at the aprosorium, but also afterwards. So we wanted to find out a way, could we conditionally inactivate it? So if we just delete the gene, it just can't infect at all. So we need a way of actually being able to allow the fungus to penetrate and then switch off the kinase. And that's what we did. So we use this process, and this is a chemical genetic approach. And this chemical genetic approach enables us to form what we call an analog sensitive mutant, PMK1AS. And the way this works is we make a mutation in a single residue, which is called the gatekeeper residue, and we convert it into this glycine residue. And by doing that, what that enables us to do is to alter the shape of the ATP binding pocket of the kinase so that it binds to a chemical inhibitor called naphthyl PP1. This is an analog of ATP. And if we now add this drug to a mutant which has this point mutation, which we can replace the native gene with this mutated version, do an allelic replacement, then we have a strain of the fungus where we can add a drug, naphthyl PP1, and it will switch off the kinase whenever we want it to. So we can inactivate it directly. So that's what we did. So we made a Magnaportha strain where we made an allelic replacement. We'd replaced the native allele of PMK1 with this analog sensitive allele. And we tested it with this drug, naphthyl PP1. So first of all, we tested it on for aprosorial development. So here's the wild type, K11, and that makes an aprosorium normally. The PMK1AS mutant also makes an aprosorium normally. But if we add the drug, if we add naphthyl PP1, then there's no aprosorium. And so that suggests then that we've switched the kinase off, and that's prevented the aprosorium forming. And that's exactly what's happened. And we can test the kinase activity, which we did, and we were able to demonstrate that that was the case. So what happens then? So inside an aprosorium, what we did is inside the cell, we allowed the fungus to infect, and this is with no drug. There's no drug at all, absence of naphthol PP1. The fungus normally fills this cell. You can see it find the pit field sites, and then it moves through, and it moves from one cell to the next. So this is what happens in a PMK1 AS mutant when there's no drug present. So now what we're going to do is we're going to add the drug and switch off the kinase. And we're going to do that after the fungus is infected. So allow the fungus to infect for 24 hours, then add the drug, and then look around at 32 hours and see what happens. So if we do that, so we've now added the drug, you can see the fungus will fill this cell. So these hyphae will fill this epidermal cell, and then they get stuck, completely stuck. They can't move out of the cell. So they are unable to move through pit fields into the adjacent cells. So the fungus becomes completely trapped by naphthol PP1. So you see a process like this. So PMK1 is absolutely necessary for cell-to-cell movement. So if there's no PMK1 present, the fungus can't move through these sites, and it can't occupy these other cells. It's stuck with and trapped within one cell completely. So that shows that MAP kinase cascade is also important for cell-to-cell movement. Now, plasmodesmata at those sites are sites of pathogen perception and sites of immunity. So the fungus is able to suppress these immunity responses by which callose normally would close plasmodesmata. So we know the fungus is able to secrete effectors at that point. And to study that process in more detail, we did an RNA-seq analysis where we took rice tissue infected with PMK1. We then added the drug naphthol PP1, and we extracted RNA from both points and did RNA sequencing. And that enabled us to show that 700 genes are significantly downregulated in the presence of the drug. Many of those are secreted proteins, and around 60 on 59 of them are temporally co-regulated with known effectors, which are able to suppress immunity. And 10 of them are a group of effectors which we study called MEP effectors. So what that suggests is that the fungus is suppressing plant immunity at those interfaces between cells. But many of these genes are also involved in hyphal constriction and morphogenesis changes that occur at that point. So to study this process in more detail, we're now doing phosphoproteomics. So we're now identifying phosphorylated proteins. In the same way, we have the absence or presence of the drug, we isolate proteins, so phosphoproteins, and then we look for targets of the PMK1 MAP kinase. And so far, we've been able to identify around 169 proteins, which are fungal proteins, which are phosphorylated during that process. And of those, we know that there are 39 which have a MAP kinase motif, which appear to be dependent on PMK1 for the fungus to move from one cell to the next inside plant tissue. And to give you an idea of the type of proteins we see, so many of them are involved in cytoskeletal remodeling. Some of them are transcriptional regulators, but many of them are unknown. We have no idea what they do. Some of them are involved in vesicle trafficking, which could be associated with the secretion of effectors. And to give you an example of what they do, so here's one of them. This is a gene called SEC31. So normally when the fungus is moving from one cell to the next, it produces lots of this protein and in a punctate distribution, and it's involved in secretion of effectors and other proteins from the fungus during infection. But if we add the drug and now trap the fungus inside the rice cell, then you can see that there are far fewer puncta, and the fungus is now trapped in the cell, and you see big accumulations of this protein. So it's misregulated with much less puncta formed of SEC31 when the PMK1 protein is inactivated. And that gives you just an example of one of the proteins that we've looked at. So how then does the fungus move from cell to cell, and what's happening at these interfaces? Well, it also involves this same class of proteins called septin GTPases, which we talked about before. So in the same way that they form a ring structure at the base of the aprosorium, they also form a ring structure, a collar structure at the points between rice cells. And if we do that same experiment again, so in the absence of the drug, these collars form at each of these interfaces. You can see there's a septin ring forming every time the fungus moves from one cell to the next. As you can see in detail here, that they form a septin ring, a septin collar. If we add the drug and the fungus is now trapped in this cell, then the septins don't aggregate correctly. And you get this diffuse septin formation, and you don't see any septin ring formation at the base of the aprosorium. So the process then is septin dependent. And of course, what we can see is there's lots of parallels between an aprosorium on the leaf surface and this structure, which we call a transprosorium, which enables the fungus to move from one rice cell to the next. And we didn't come up with this name. This name was coined way back before I was born in the early 1960s by a group of sharp-eyed mycologists who said they could see swollen structures inside plant cells being infected with the fungus. And they coined the term transprosorium. And I think they were correct, that this is actually a specialized infection cell, just like the aprosorium, with many parallels, including the formation of septin-mediated actin relocalization to enable the fungus to move from one cell to the next, but at the same time as actively suppressing immunity as it's doing so. And then finally, you can see this model for how the cell works. So the fungus, the aprosorium, has penetrated inside the rice cell. The PMK1 kinase is involved in forming, regulating effector gene expression. Those effectors will suppress host defense. At the same time, it's also involved in cytoskeletal remodeling, enabling this septin collar to form and enabling the fungus to move from one cell to the next. So forming this severe hyphoconstriction and enabling the fungus to move from one cell to the next cell. So to summarize everything I've told you then, the fungus forms this special cell called an aprosorium to infect rice plants. That depends on septins, also on the cell cycle, on autophagy, and on a special kinase pathway regulated by a protein kinase called PMK1. There's another sensor protein called SLN1, which senses once TERG has reached a threshold and then enables the PEG to form. Tissue invasion requires a huge battery of fungal effectors, and they suppress immunity. And the fungus then makes an infection structure called a transprosorium to move between rice cells. And the PMK1 MAPK actually regulates both of these processes, aprosorium and transprosorium formation. And the people who are involved in the work, I won't mention all of their names, but many of them are the most important in the work that I told you about. Was Lauren Ryder, who did the work on SLN1. Miriam Osses Ruiz did all the work on PMK1 and on transcriptional regulation. Neftali did all the work on phosphoproteomic analysis, along with Shah and Bozeng and Vincent, who worked on effectors during this process. And Iris works on effectors now, and Alice did a lot of the movies and microscopy that you saw as well. And I've got great colleagues at the Sainsbury Lab who's helped me, particularly my proteomics team, Frank Menke, who's a great colleague, and his team, Jan and Paul, who have helped us with a lot of that mass spectrometry analysis, and also the informatics team and my great colleagues at the Sainsbury Lab. So I'm happy to answer any questions that you've got, and thank you for listening.
Thank you, Prof. Talbot. It's really great presentation, and I really enjoy your visual presentation as well. And we have already had many questions here. Okay, in the chat. Okay, I will read for you. Yeah, first from Ibu Sophia Lena. I am Sophia Lena from Mula Warman University. My question: Blast is an endemic disease in rice plantation in Indonesia and can cause loss of production until 20%. Since you worked a lot on the fungus, how can we manage the pathogen effectively using biotechnology methods? And the second question, since the fungi are very easy to adapt to the host or invade and grow rapidly in the host tissue, are the fungi of Magnaforte oryzae easy to mutate? So,
well, the answer to the second question is yes, they are, and this is one of the problems. This is why rice blast is so difficult to control, because it forms a big battery of effectors. And every time we try and deploy a new resistance gene, which will be encoding a receptor that will identify a given effector, all that the fungus has to do is mutate that effector, and it will then become virulent again. And because it has a whole battery of 400 effectors, it's able to do that normally without losing too much fitness, it seems. So therefore, it's able to overcome resistance, which is why we know as plant pathologists that rice blast can normally, within 1 or 2 growing seasons, will overcome a new resistance gene very rapidly. So to answer the first question, how would you control blast then? So the way that we're trying to do this is by stacking resistance genes and different resistance specificities. So the work that we're doing in Africa, we monitor the population of the fungus, work out the effectors that are most prevalent, and then we deploy appropriate resistance genes. But we normally do that and this is working with IRI in the Philippines. And we normally deploy 3 or 4 different resistance specificities, normally 2 to 3 major genes, then also a recessive resistance allele like PI21, which is much harder for the fungus to overcome, and also some form of polygenic resistance, a QTL type of resistance. So if you can introgress all of those into an elite cultivar, then you can come up with something which is quite durable. And those are the ones that we're testing in field trials. So I think that that's one solution. I think other biotechnological solutions could involve things like gene silencing, or of some of the targets that I've shown you potentially, and also methods where if you can identify susceptibility factors, then those could be addressed in rice using CRISPR gene editing techniques. So I think there are a number of ways that can be explored. None of them are easy, and all of them require normally multiple different genes, because otherwise the fungus will simply mutate and overcome them. But I think there are means to do this using biotechnology. We're very enthusiastic about doing this, and we work quite a lot on that type of work.
Okay, thank you for your great answers. Okay, we will move to the next question from Bapak Muhammad Iqbal Maulana. Prof. Talbot, I have question. When the fungi are inside in the plant cells or even destroy the plant cell walls, doesn't the plant receptor recognize the hyphae as PAMPs? What hormones or compound the fungi uses to manipulate plants' defense? So about the response of the plants.
So I think the first answer, the first question is that I think that as the fungus is actively growing, it's always in living cells. So that the way it's able to suppress PAMP-triggered immunity is because it's in living cells and continually secreting effectors which suppress those processes. So we know that it can suppress chitin-triggered immunity, for example, in the apoplast, also glucan-mediated immunity in the apoplast, and then internally, it's suppressing a whole raft of different functions involved in pathogen perception. But of course, when those cells, when it destroys the plant cell wall, I think at that point, it's also killing the plant cell. So I think that it's a sort of classic biotrophic necrotrophic switch. So as the fungus is growing biotrophically, it's suppressing all of those processes. But once it's actually moved from that region, the cells that are left behind, I think, normally will die, and the fungus will then use all of that nutrient to fuel sporulation. But at that point, they can't, the plant can't launch a PTI response, a PAMP-triggered immunity response, because it's those cells are dying or are dead. And then the second, hormones and compounds, well, it secretes a lot of effectors, I say more than 400 effectors to suppress immunity, but also lots of metabolites too. So there are lots of small molecules which also affect plant cells. So some of them For example, there are pericalacins, which will alter actin organization in plants. There are lots of metabolites that do this, and may also even suppress things like phytoalexin production. So many of those we don't understand, but there are many secondary metabolites as well as proteins being produced.
Okay, thank you. So complicated for the infection process. Okay, and then next we move to the question from Prof. Lucas Susanto. Can the blast fungus infect indirectly through stomata? How is the mechanism? As you said that after the fungus infect, the fungus needs 50 minutes to spread in the leaf tissue. What is the main factor influencing the spread? Is the same time 50 minutes for several physiological rays of the fungus? What is chemical compound produced by the fungus used to invade the leaf? Thank you. I think the chemical compound you have already mentioned, so we can one by one answer. So how about the infection through stomata?
Yes, so let's go through all those questions. So the fungus normally doesn't use stomata. It normally doesn't do that. It normally just smashes straight through the wall. So it's quite interesting. In fact, normally it will, if you look on a leaf surface, you can see it go almost round stomata. It doesn't use them. It seems to ignore them and just go through the wall. So it's not like rusts or powdery mildews or other fungi that use stomata. So it doesn't do that. It just uses physical force to break the cuticle. So it needs, so that's it's 50 hours rather than 50 minutes. So it uses it. So after about 50 hours, the fungus, that big field of view I showed where you've got lots of fungal hyphae, that's around 2 days after infection. So about 24 hours to infect and move into the first epidermal cell. 30 hours or so, it will move into adjacent cells, and in 50 hours, it's filled a lot of tissue. That's very, very fast. I mean, it's really very fast. The main factor influencing the spread, well, I think there are probably many factors which probably are associated with effector production, suppression of immunity, but also many fungal factors, for example, the ones involved in cell-to-cell movement. Also, the thing I'd really like to understand is how does it make its way to the vascular tissue? Because it seems to do that very quickly, and we don't really know how that's done. So I'd really like to understand that. And I say the chemical compounds, there are many of them that are used in the invasion of leaves. The best known ones are associated with affecting things like actin organization, the pericalacins, but also even things like tenulzonic acid is produced by blasts. So it will produce things which are actually affecting and suppressing plant defense responses, things like the PAL pathway and phytoalexin production.
And do you think it's already also same for the different race of Magna Forte Orizae? Because you know that there are so many races of this kind of fungi.
Well, from the sequencing that we've done so far, they are all very different. So, and they differ in that effector repertoire. So very often you'll find that if we look at the whole pangenome of rice blast, so try and pull everything together from every different form, then we're up to more than 800 effectors. So there are lots and lots of different ones being produced, and also lots of different secondary metabolic pathways. So some of the different races and host limited forms have very different secondary metabolic pathways. So I think that there is a lot of variation in many of these processes.
Okay, thank you. Then we will move next. Question from Ibu Liania Sartika. Dear Professor Talbot, I am Liania Sartika, a double degree student of University of Brawijaya and National Taiwan University. I have an experience regarding to the rice blast and millet blast. In my lab, we have several rice blast strains isolated from the rice. Interestingly, it also can infect the foxtail millet plants by spray inoculation. So in this result, I might conclude that probably the foxtail millet is the secondary host of the rice blast. And another side, we all know if we have rice blast, millet blast, wheat blast, with its specificity, from the experience that I have, do you think that rice blast has evolution chance to invade other hosts and how it can be happened? I mean, does blast has an ancestor that can be infected many hosts? About the host range, Prof. Talbot.
Yeah, so I think that's a great question, and I think the answer to some of this is probably yes. Now, I mean, a few years ago, I would have been less sure, but actually, I think that we're seeing the same thing. We can see we can actually see fungal strains which are also able to infect Soteria and rice. You know, we're seeing some which affect different grasses. And of course, farmers for many years have seen lots of rice blast type lesions in the field margins and seen, and they always suspected that there was movement between grass hosts and the fungus, but we didn't really have the tools to study it. And where it was looked at primarily, most of the studies back in the 90s suggested that rice blast populations were really very, very different. I think now with sequencing, we're actually seeing that there probably has been hybridization between these different forms. And some of that could be sexual recombination. They may be interfertile, but also some of it could be happening through parasexual genetic exchange. So maybe happening through anastomosis and heterokaryon formation and parasexuality. So I think there is certainly scope for host jumps because we've seen it happen in wheat blast, and the nearest neighbor is a Lolium or an Echinocloa pathogens. There may have been more than one host jump. So I think that yes, there's a possibility of there being a host jump. In terms of ancestrally, I think that the short answer is we don't really know. But I think if we look at the origins of that whole group, the whole genus, then they infect so many different grasses and then have radiated into these different host-limited forms over time. So. And certainly at the moment, almost all blast pathogen strains will infect barley, for instance. So barley seems to be like a universal susceptible host. So I think that it's quite possible that an ancestor was able to infect more hosts. But we don't know is the answer. But I think that your speculations are probably going in the right direction. I think they're probably correct.
Okay. So, Prof. Talbot, we still have many questions, but no worries because the committee also told me that the discussion time will be extended until 3:15. So we still have time. Okay, we will move to the next question from Dr. Shibli from Brawijaya. I would like to know about your fungal mutant collection that you are working with. So after particular gene deletion process, is there any character changes after the deletion, such as in complete conidia, the conidia become major, but they only have one cell, not 3 cells?
Yeah, so among all of our mutant collection, we certainly do have some mutants that have some of those characteristics. So we check all of our mutants for all of those different, we check them for conidiation, for growth on different carbon sources. We check their fitness. We do lots of tests on them. I mean, and many of them have just specific effects on virulence, but others have effects on sporulation too. So a mutant like the one you're talking about, the one that's probably closest to that is a mutant called SMO. The SMO1 mutant, which we published a couple of years ago. And that forms round conidia, which are normally 2-celled. And they cannot form aprosoria properly, and those aprosoria can't function properly. So it's essentially non-pathogenic, very reduced in virulence. So SMO is an example of that, which affects spore morphology. And there are a number of other mutants, which were called con mutants, isolated by another group many years ago, which also are all affected in conidial morphology. So there certainly are some, and some which are reduced in the number of cells. I don't think I've ever seen one with just one cell. I think I've only seen 2-celled and 3-celled. I don't think I've ever seen a single cell conidium. But yes, there are mutants that have some of those characteristics.
Okay, thank you. The next question from Dr. Lukman, also from University of Brawijaya. I have 2 questions. You mentioned that rice blast fungus release effector during infection into plant cells. What kind of effectors and their function during infection? And the second, you also mentioned that hyphae of Magnaporte oryzes breed to other cells via plasmodesmata. So why through plasmodesmata?
Yeah, so good question. So in terms of the effectors, there are so many of them and many different functions. I can give you a couple examples. So there's an effector, for example, SLP1, which is a protein, which is a chitin binding protein, and it will bind to chitin monomers at a higher affinity than the CBIP chitin receptor in rice. So therefore, it prevents that immunity response occurring. Inside plants, there are many effectors. For example, AVR-PIK, which is recognized by the PIK resistance gene. As an effector, it actually binds to heavy metal associated domains in proteins, which are involved in plant defense. So it can't mount, the plant can't mount a reactive oxygen species mediated defense response if those proteins are being perturbed by these effectors. And it seems like many effectors bind to HMA domains. And there are many more. There are so many. One we're studying at the moment is a chloroplast effector, which will go to the chloroplast and bind to part of photosystem 2, and will affect the ability of chloroplast to mount an immune response with ROS. So many different proteins and many different mechanisms of use. And we're only scratching the surface. We probably, the whole rice blast community probably knows the function of maybe 10 effectors, maybe less, maybe 6, 7 effectors. And we know there are more than 400. So most of them we don't know what they do yet, but we will do. Now, why do they use plasmodesmata? Again, I think that what they're doing is that when they move from cell to cell, they are able to identify an indentation topologically. And they know that that's a softer point because that's where there are lots of plasmodesmata. And that's probably why those regions are being picked, because it's easier to move through that region than elsewhere. The other reason, though, of course, is because it gives you a mechanism to maintain membrane integrity, because it's a conduit where you've got cytoplasmic continuity. So if the fungus is surrounded by a plant plasma membrane as it moves through, it may be able to maintain the integrity of the membrane as it moves through that site. So maybe that's why it picks those particular regions, but it does seem to use them preferentially to anything else. I mean, it may well be that it has to use them, but it certainly, we can't find much evidence that for when it doesn't use them. It seems to always use plasmodesmata.
Okay, thank you. And we will move to the next question from Jauhar Shauki. So my name is Oki from UB, following Liania Sartika questions. I think it's about the host range. Just wonder, any other host specificity gene like PWL, which specifically prevents rice blast to infect rice? Thank you. Is there any?
Well, there must. I think there must be. So PWL2 so the PWL family are involved. They're host specificity determinants for weeping lovegrass, Eragrostis curvula. So that's a family where the presence of PWL2 prevents it from infecting weeping lovegrass, which means that weeping lovegrass must have a resistance gene for rice blast, which is able to identify that effector. So we're working on PWL2, and we've actually, because there are multiple copies in cells, it was impossible to get a mutant, but we've used CRISPR now. We've knocked all 3 copies out in rice blast, and we've shown that there is actually an effect on virulence. So it clearly has fitness consequences in the fungus too, but it does verify all of that work on weeping lovegrass. So I think that actually there must be genes, and they must be in foxtail millet and in lolium, in all of those other. So I would say it's probably in all of those different grasses. You know, in perennial ryegrass, in torpedo grass, in all of those other grasses, there must be resistances to for example, the rice blast variety, the rice blast races, and therefore you're right. There must be host specificity determinants. There must be effectors which are recognized just by those grass hosts. If we could identify them and clone them and put them into rice, we'd have a really good way of controlling rice blasts, which is why we're studying PWL2 for that idea. So yes, we're thinking along the same lines that you are about that.
Okay, thank you. Next, we move to the question from Endah Yulia. My questions: how is the typical blast symptoms, like spindle shape or spindle shape wide in the center and pointed towards either and formed, does it relate with the development direction of the hyphae inside the plant tissue, or related with the morphological characteristic of the rice plants? So the specific of the symptoms.
Yeah, again, that's a fascinating question. So I think that I think it probably must be associated with that. It must be associated with the way in which hyphae develop, and I think that they must be growing in that longitudinal axis more than laterally to form those longer lesions, because it's absolutely characteristic of blast lesions. So how those lesions are spatially delimited, we don't know. So it's possible that that is part of a more systemic plant defense response, but that conditions it as well as fungal growth, but we don't know. I'd love to know the answer to that question of why you see a particular shape. I mean, it'd be quite interesting to screen for mutants which had a different shape. So maybe we could address that by screening for those different lesion shapes to try and answer that question. But I think it probably must be associated with, as you say, the development and the predominant direction of hyphae inside plant tissue.
Okay, thank you, Prof. Talbot. Okay, next question. We also have a nice question from Sri Widinugraheni. Do you know that PMK1 also presents on other Magna Forte infecting strains, such as on Sigatoka disease? And will they have the similar mechanism during infection?
So yes and no. So in fact, many pathogens have got a PMK1 homologue. So there is a homologue to PMK1 in definitely in Sigatoka. So they have it. So does Panama disease of banana too, Fusarium, Oxysporum, and so does Mycosphorella graminicula on Zyma septoria on wheat, and so on. So actually, there are most pathogens where it's been looked at have a PMK1 homologue, and if you knock the gene out, it's non-pathogenic. So it's very conserved. It's a conserved pathway for invasive growth. Some of those fungi make aprosoria and some don't. So even ones which don't make aprosoria, clearly their ability to invade plant cells is conditioned by PMK1. So I think PMK1 is part of an ancestral pathway associated with invasive growth. And it's homologous to in yeast. There's a gene called FUSE3, which conditions pheromone signaling, but also conditions invasive growth even by yeast. So when it forms pseudohyphal growth. So I think it's part of a quite a conserved and ancient pathway which is involved in invasion. So yes, there are PMK1 homologues in many of the pathogens that we all study. Yeah.
Okay, thank you. And then we move to the question from Ali Wafa. As from your presentation, what that mean we must reduce the PMK1 activity or inhibit them to tackle Magna Forte oryzae infection process, and how we must inhibit them, like by using chemical or any compounds outside to develop the resistant gene house. I think based on complex enzyme or protein activity during the infection process, become harder to build a resistant host. What do you think?
Yeah, very, very good. And I think it's a very good question again. So I think what's interesting there is that you're sort of highlighting how difficult it is to control plant disease. So if we could maybe develop chemical inhibitors for PMK1, for instance, it will be a good target. The thing is, of course, is that kinases are actually so promiscuous that finding a very specific kinase inhibitor is really hard. So the way we've done it is by making a mutation in the gene, which then enables it to just target one kinase. To do that to develop a new fungicide which only affects one kinase is really hard, and of course could also be subject to the kinase mutating and overcoming the effect of the inhibitor while still maintaining its activity. So I think that there are a number of ways of doing this. I think that actually potentially gene silencing approaches might be a way of actually suppressing that activity, because it's such an important activity. But I don't think it could ever be done singly. I think you'd always need to try and identify at least 2 or 3 different targets and have a chemical inhibitor or suite of chemical inhibitors with several targets, or preferentially, because of course we want to try and reduce chemicals because they're all coming from fossil fuels, from oil production. So if we want to try and do something more sustainable, it will be better to use genetics. But it's the same thing applies, that we can't have single gene resistance. We've got to try and stack things in groups to try and get more durability and different forms of resistance. So I think it's like the answer I gave about the biotechnology question. I think we've got to do things in complexes rather than single targets.
Okay, thank you, Prof. Talbot. I hope you're still happy to answer. We still have 5 minutes. Is it okay? Yeah. Actually, still there are so many questions in the chat box, but let's finish until the rest of 5 minutes. Okay, next from Muhammad Junaid. Thank you for interesting slide, Prof. Talbot. Blast disease in our region is endemic, and no doubt the pathogen is very clever to undermine plant-resistant mechanism. In what way we can develop practical technology to help farmer against the pathogens?
Okay, I think one thing that would be very good is to try and monitor the blast population. So if you can isolate many isolates of the fungus from different regions, and then inoculate them against the international differential set of rice blast isolates, you should be able to identify the resistance genes that are able to exclude the majority of the endemic population. So you use pathogen-guided breeding. I think if you do that, you would be able to determine which are the major PI genes which are giving you, which should theoretically exclude the majority of the population. And then you need to guide breeding programs to introgress them in groups, not just one gene, never one gene, because that's just going to be another epidemic, but in groups. And also consider recessive resistances like PI21, which is so good at and relatively durable, and always introgress that at the same time. That's harder because it's recessive to do the breeding, but it's I think those are practical solutions. I think the other thing is to think about if there is access to fungicides, it's really advising them on the best part of the growing season based on prevailing weather conditions to spray. But so providing that type of real practical advice is also good. But I think that in terms of a way of durably controlling the disease, the best thing is to know the pathogen population. And that can be done using pathotyping. It doesn't need to be done using molecular biology, although if you're interested in doing that by doing sequence analysis and more elaborate analysis, then you should get in contact with me, because I'm sure we can help do some of that.
Okay, thank you. And then the question from Prof. Ayu Parawansa. Prof. Talbot, very interesting presentation. Can fungi mutate with the current climate change? So the fungi relates to climate change. What do you think?
I think that they are doing that. And the only evidence I have for that is because we're beginning to find Magnaportho species at more northerly latitudes than previously. So I think that so recently one of my colleagues, Sofian Kamoun, has been monitoring blast populations across Europe, and they're beginning to find things further north than one was expecting. So I think there is some evidence this is happening. I think we'll know more in the next few years, and I think my colleagues group will analyze those strains and sequence them and try and work out what's happening. But I think that we're beginning to see that. We're also seeing that from growers both in the northern and southern hemisphere. We're seeing the fungus moving towards the poles gradually. We're seeing it move away from the equator. So I think that I think those processes are happening, and it will be a very interesting experiment to actually look at temperature optima for growth and see if it's changing in lab experiments, because I think that we are seeing that as well.
Okay, thank you, Prof. Prof. Talbot. We still have 1 minute, so I will choose one short question about the silica content in the epidermis of rice leaves. If we increase the content of silica, for example, the silica fertilization, it will help?
Yes, it definitely will help. There's actually good evidence for that. So so there's some really good evidence that the world expert on this is a guy called Lawrence Datnoff in the US, and he's shown that rice lines with increased silica content are more resistant to blast because they are reducing the frequency of Aprestorium mediated infection because it's harder. So yes, of course, you've got to look at that in the context of what it does to yield, and you've got to balance it against yield and test it effectively in terms of other grower traits, rice traits. But yes, there's a link. There's definitely a correlation. There's a lot of literature on that too. So yeah.
Okay, thank you very much, Prof. Talbot. So our time is already finished now, and so thank you once more for your insightful presentation and also quite a long discussion. I think from your presentation we learned a lot about the complicated of the pathogenesis process of Magnaphorte oryzae, especially the role of Apresorium and Transpirosorium and PMK1 MAPK in the pathogenesis. And we really would like to express our gratitude, and I think the committee has also prepared a wonderful gift for you, Prof. Talbot, for giving us the great presentation. There is one local artist here that, this is for the certificates and also the painting for you, Prof. Talbot. You see here, you explain about the Magnaphorte oryzae in the first slide, I think. Hello?
Okay. I can't see.
Yeah, this one.
Can you share
see?
this? I can't see it. You have to share the screen, I think. If you share your screen, I may be able to see it. Thank you.
I don't know how to share, but it's already in our screen actually. Okay, I think the committee will share to you afterwards. And
thank you.
Thank you very much. And then for the previous.
Oh, I see. Thank you. I can see it.
You see?
Wow.
Yeah, very nice.
Thank you so much.
Yeah. You see, the face is really you.
That's lovely. Yeah. Thank you.
Yeah, it's nice. Okay, and the next also the certificates and also the painting for Dr. C.V. Indarti, the previous speaker. Yep, here you are. So, okay, would you please slideshow for the painting? Hello, Bu Siwi. Bu Siwi, are you still there? Okay, I hope Bu Siwi happy to accept. Wow, nice presentation. Painting, and also I love the Indonesian flag here. Okay, thank you very much. And once more, I would thank to you, Prof. Talbot, and also Dr. C.V. Indarti for the very important presentation, and also for all the audience for the active discussions. And after this session finish, and it means that all of the afternoon session also finished. So thank you very much, and we do hope that we will meet you all soon, and also welcome someday in Indonesia, Prof. Talbot. Yeah, it's very nice, to have you here. And so thank you very much again for all audience, and I give back the session to the Master of Ceremony. Thank you.
Thank you so much.
Well, thank you very much to Miss Ani Widyastuti, PhD, for leading the second conversations. Looking forward to seeing you again on tomorrow event, the second day of this conference. Well, ladies and gentlemen, that's all the keynote speaker session. Thank you very much to all speakers, moderators, participants, and also the invited guests that participate in international conference and the 26th Congress of Indonesian Phytopathological Society with the theme, the role of phytopathology in realizing healthy plants towards sustainable agriculture in the Industrial Revolution era 4.0. Ladies and gentlemen, unfortunately, we are already in the last session of this event itself. So we would like to extend our gratitude to all the distinguished guests and on their presence and also the participation in this event. Now, let us express our gratitude to all parties that really support this event. Thanks to PT BC International, PT Petrokimia Kayako, PT Bank Sentral Asia senantiasa di sisi Anda, PT Pupuk Indonesia Persero, PT Medion Pharma Jaya, Krisan Catering, PT Multikarya Transcendent, dan Grafika Advertising Malang. We remind you all that tomorrow we still have an event. There will be a paper presentation, announcement of the winner of Plant Protection Competition 2021, and also the closing ceremony. For tomorrow agenda will be held on 7:30 AM Western of Indonesian time, and also it will be live streaming from Perhimpunan Fitopatologi Indonesia YouTube channel. Looking forward to seeing you all again on tomorrow event, the second days of this conference. I'm Khairunisa signing out. Thank you very much and see you again. Well, there is an additional information for the international conference participants. You can leave the room on this Zoom meeting, but you can join again tomorrow event on the second days of the conference. But for the Indonesian Phytopathological Society or Perhimpunan Fitopatologi Indonesia members, please stay here because of this we will have and directly continue to the 26th Congress of Indonesian Phytopathological Society, and I will guide the event in Bahasa. Thank you. Assalamualaikum warahmatullahi wabarakatuh. Salam sejahtera untuk kita semua. Shalom, Swastiastu, Namo Buddhaya, dan salam kebajikan. Yang terhormat Ketua Umum Perhimpunan Fitopatologi Indonesia, yang kami hormati Ketua Pelaksana the 26th Congress and International Congress of Indonesian Phytopathological Society, dan yang kami banggakan hadirin dan tamu undangan yang berbahagia. Puji syukur kehadirat Allah Subhanahu wa taala karena berkat rahmatnya kita dapat berkumpul pada hari ini dalam keadaan sehat walafiat. Dan saya Khairunisa selaku Master of Ceremony pada Kongres hari ini mengucapkan selamat datang di acara Kongres ke-26 Perhimpunan Fitopatologi Indonesia. Baiklah, memasuki acara berikutnya yaitu menyanyikan lagu Indonesia Raya, kemudian diikuti dengan menyanyikan lagu Mars Perhimpunan Fitopatologi Indonesia yang akan dipimpin oleh Ibu Dina Agustina selaku dirjen kita pada hari ini. Kepada beliau disilahkan.
Menyanyikan lagu Indonesia Raya.
Baik, kami ucapkan terima kasih kepada Ibu Dina Agustina yang telah memandu menyanyikan lagu Indonesia Raya dan juga Mars Perhimpunan Fitopatologi Indonesia. Baiklah hadirin, untuk memulai kegiatan kongres hari ini akan dibuka oleh Ketua Perhimpunan Fitopatologi Indonesia. Kepada Profesor Dr. Insinyur Abdul Latif Abadi MS, disilahkan.